Hello everyone, welcome the heck in. Happy Monday fun day, y'all. Chat, who is not ready for it to be Monday? Who was not ready for it to be Monday? I woke up this morning because Lita was waking me up. Because the little one was ready to go. You know, she was awake and she's like, it's dad time, let's go. And I was so thrown off. Look at what amazing stuff Carla's doing. That I had no idea. I was like, are you sure it's... I'm like, are you sure it's Monday? She's like, yeah, it's Monday morning. I'm like, oh God. I'm not ready for it to be Monday. I'm not ready for it to be Monday. Oh! I got this cool looking thing in the mail. It got better at Quaff. No idea what you got, but I'm glad it was cool. I'm glad it was lovely. I'm glad it was amazing. Carlo! Why the NTS? Is that like... What is that? The stuff that you use when you can't use money? Right? Instead you use these fancy pants like these extra things. I don't know, Carlo! You're talking craziness! Carlo! Chat, welcome on in. First today was Smikes. Y'all, Smikes has been first today for like 3 weeks now. As an NFT, yes, the NFT. Think dubstep? No, Carla, the only thing I know about dubstep is that if you're taking a bath, that's when you listen to it. You listen to dubstep when you're taking a bath. Or tubstep, for that matter. Oh! Now I understand. Carlo, now I understand. Now I understand, Carlo. Now I understand. It took me wanted it to be not to be Monday. It's Bubbles, hello. Laura Gale, hello. Woosa, hello. Carlo, the stream scarf is in his house, making some live letter art with us. We also, let's see, have Coffee Rocks. How you doing, Coffee? Welcome on in, Coffee Rocks. The breathtaking Debs in the house. Peekaboo Draws, hello, hello. Gwendolyn P. Gwen suggesting that Carlo is aged. It's terrible. Terrible, absolutely terrible. Coffee Rocks! With the dancing Kevins, and then, oh my goodness.
Don't you like me a little bit?
Ew, I don't even
Big
like
Gamey.
you a little bit.
I heard you like me a little bit.
Hi, Big Gamey. I also see Carlo was
Just
naughty
a little bit.
and said sorry, so I've deleted his comment. He said the word sorry for needlessly, so it's been deleted. Fall Machine sneaking up on in here. I caught him though. Andrew CZ, the breathtaking soul that is Andrew CZ. Tokatao, hello, Tokatao. Tech, first time chatter. Tech 2674, welcome the heck in. How are you doing today? Big Gamey in the house as well. Big Gamey, how are you doing today? Becca's Crafty Life, how are you? Little Oxide, hello, hello. How are you doing today? Dackham, hello, hello. Welcome on in, everyone. Blink too long on Friday was Monday? I think so, Big Gamey. I think so. Quath the Raven, this guy's jamming out some drums. Quath, did you get Kevin on some drums today? Did you get some Kevin on the drums? Becca almost done my first commission piece. Let's go, Becca. Becca, what was the first commission piece? What did you make, if I may ask? Tolkien Tao, how are you doing today? Welcome in, everyone. Guys, we got a couple more shout-outs to do. We also have the lovely Becca's Craft Life, who is near complete with her very first commissioned piece. Crocheter, knitter extraordinaire is Becca's Crafty Life. And then we'll also have the lovely Bits Gamey as well, which we love Big Gamey. Stitch from Lilo Stitch. And are you doing any stitching on him, Becca? I am so sorry, Becca. I am so sorry. That had to be done. That had to be done. I'm a little bit silly willy today. I may have Monday brain. I did get some work stuff done that went... It was alright. It wasn't anything bad. I was just very faded. It was bad enough where I was sitting with baby Lona and near fell asleep. Like, we were just... Then I had meetings this afternoon, too. We did go on a walk today, and then the afternoon meetings, I was like that. I near fell asleep during the meetings. Unfortunate, but what are you gonna do? Twitch Addict, hi Twitch Addict, welcome the heck in. Chat, we have a couple of things to announce. There is a new command for subs. There is a new animation that we have in there. So if you'll see, it is in the commands of the new command list. It is there. This is the piece that we did yesterday, the 3D printing Antonia with Kevin. Dancing celebration in detail, chat. Then they go get a commission. Becca, it gets going, and then there's the next one, and the next one, and the next one. There we go. Coffee Rocks, thank you for triggering the 3D printer command. Now we have a 3D printer animation too. And the next one, of course, Gwendolyn P is like, well, Balint, did you get haircutted today? Gwen, no one gets haircutted on a Monday. So chat, for 2,640 channel points on the line, the correct answer is no, absolutely not. We did not get haircutted today because it's a Monday. No one gets haircutted. When I woke up thinking that it was not Monday, it was not about to happen. It was not about to happen. I am for sure orange sometimes, Carlo. Over months, if I got one of the boop. Becca, it's the hustle, man. We gotta... I don't know the best way to do it, but I'm sure there's like how to reach out to people. Putting your name out there. Putting your work out there. And then eventually, stuff catches up. And you start getting more and more of those. It's a slog, Becca, but... Hey, first one's the hardest, and then it's just keeping up the energy there. Chat, what are we working on today? What are we looking at today? We are going to be doing two things. Yeah, thank you for the Lord, Yan. Got more shout outs. Please go follow Yan. He's live right now. If you go follow him right now, he might jump a little bit. He might jump. Got a little bit of a sound alert there. Gamer extraordinaire over in the UK. Go check out the amazing Yan, folks. You'll never be disappointed.
Ah, Gucci.
He is very Gucci. Devs, he is very, very Gucci. Chat, go check out the lovely Yan. We will be today doing a couple of things. One, so we had a freeze last week. Over Saturday night, Sunday morning, we did have a freeze. And so I did not bother collecting any pond water today. Because I'm like, you know what? It's far too close. Given how few critters we found last week, I was not hopeful of us getting any good pond water samples today. You had one last night too, Quad? Yeah, it's... Now I have my windows open. Quad, that makes absolutely no sense. So, I didn't grab pond water this week. Instead, I grabbed tree bark and the lichens and the mosses that are growing on the trees. Why did I do this? There is usually a distinct possibility that there are tardigrades swimming around in those samples. So we're going to try to find some tardigrades. Now the best way to find them is to take the lichen and moss samples and put them right into water and let them soak. Once they are done soaking for an hour or two, you can take out the samples and see if there's any tardigrades in there. So what I actually did was this afternoon, around 5, I started soaking the samples. And so is there any tardigrades in here? We might be able to catch some now. If we don't find any, I'll clean the dish. We'll soak new samples while we look at some amber. I also, Gwendolyn P will be excited. We have 2 more Cretaceous amber samples. So these are 90 million years old. They do have insects in them. I haven't looked at them yet, but we'll take a look together. And then we have our Dominican amber samples as well. All while Carlo is going to be doing some lovely live letter art above my head. So that is the kind of on the plan for tonight. Later on this evening at 11 p.m. Eastern, Nerdowino, I'm going to be on Nerdowino's stream, and he's going to be doing an interview, chatting with me about SciAnts and both SciAnts. So that is the plan for tonight later on. So I hope you guys will stick around for that. Chat, one more shout out for the Fancy Pants BitGamia, who's in his house. Guys, if you're not checking out BitGamia, single greatest father on the planet, game dev extraordinaire, and just a legendary soul. We love BitGamia here. Chat, make sure to go follow BitGamia. Moo Hoodles, you can't. If you would have come out 30 seconds earlier, then I would have countered with Moo Hoodles. That sounds cool. Tardigrades are also cool. Nerdweed is also cool. But you know who else is cool? Moo Hoodles is really cool. That's would have been the logical flow of it, Moo. But since you came out 30 seconds too late, now I can't do that. Now I have to sit here and wait a minute and 30 seconds before I can make that commentary. Moo. I just want you to think about what you just did. Moo Hoodles. That you totally threw off the groove so that I can shout you out and make it sound fancy. Okay, Moo? I want you to know. I want you to know that, Moo. It's not fine, Moo. It's not fine. I should have been on point, Moo Hoodles. I should have been on point to make it sound fancy and like totally planned out, but instead now we're just filling it with awkward talk and awkward silence. And you know what? I guess it's fine, but at the same time, it's just silliness. Go on. I know, beware of the groove. You're off the Emperor's Groove, Quoth the Raven. You're off the Emperor's Groove. Sorry, Purge. Just purge my message to come again if you... No, Moo Hoodles. No. It's been done. It's too late. It's in the VOD now. It'll be on the YouTube page with the lovely Debs. Does all the YouTubing, so yeah, make sure to check out the YouTube page as well. Man, I lovely and amazing Debs. Debs, we love your face. Music Girl and Grimly. Our two breathtaking moderators. Hello. How are y'all doing today? Welcome on in. Quoth, should there not be a live-action remake of The Emperor's New Groove? As long as they get the same actor to do Kronk. Actually, they could probably get David Spade to do it as well. Although David Spade is really... He's on the older side now and probably wouldn't work for the young Emperor. But they should have the same actor to do Kronk. And they should still mention spinach buffs. If y'all know what I'm talking about, then you're in the absolute right place. I love Emperor's New Groove. Instead, Quoth, they're remaking Moana as a live-action movie. Don't get me wrong, I loved Moana, but it seems... Cliff, hit it! Hello, Cliff Alistair McLean. How you doing, Cliff? Eartha Kid is no longer here. Yeah, exactly, Grimly. I have to go not yell at Jimmy on stream of how to set up in the background. Music Girl. You take care of Mr. B. You take care of Mr. B. The poison, the Cusco's poison, the poison for Cusco. Quoth, spinach puffs, Kronk, absolutely essential. Needs to be in the movie without any question. Now, the live-action Lilo and Stitch Little Oxide, I'm okay with. Because it's been long enough, and I don't mind the Moana remake either, but I read they're doing it because they are getting a good deal on filming in Hawaii with back-to-back movies. I know, Smikes, I know. It's a little bit silly pants, but what are we gonna do? That and his theme song are the two main things, well, the cat and Yzma. Yes, Quoth, that's true. That's very true. Gwen, we do have the ants moving.
Uchi.
There's a couple of things I want to show y'all. Gwen actually redeemed this a little bit before the stream started. Gwen, you can see here again, their trash pile is growing. They're still bringing out stuff. I did another peer through in there. Couldn't find the queen's body again, so if she is gone, I'm not sure. I'm not sure if she is gone or not. I mean, I saw the body. And she wasn't responsive, so I assume the queen is lost. But there is still that second queen, maybe. Quoth, not even leafcutter ants, but most ants. Most ants. There is a trash job. Leafcutter ants are so specialized, there's an entire cast devoted to taking care of the garbage in the colony. Trod an anthill today by accident, they were active after that. Did they bite you, Big Gamey? Chat.
Hello there.
Speaking of biting...
Hello there.
Make sure to follow Jomio. Jomio. Wherever art thou, Jomio? Deny thy father and refuse thy name. Jomio is Jomio. Chat! If you're not checking out Jomio, please go check out Jomio. Legendary artist extraordinaire. Jomio, listen. I had such a laugh this weekend. Chat. Jomio, first of all, a breathtaking artist extraordinaire. Built like a Greco-Roman god, just like our friend Andrew CZ. But he's a big Manchester United fan. He's a big Manchester United fan, folks. What's green, folks? Long-time follower, first-time chatter, followed you because of the kitties you post on Discord. Oh, thank you, Had. We have 4 cats. And I did turn them into emotes, so we do all of our own artwork here. And so there's Noodle. There's Barley. There's Cannoli. And there's There's barley. Or the hops, cannoli, and then barley. They are brothers, and I get them confused sometimes. And then, of course, there's Noodle doing a head bobbing, and this is her meow. That cat is my absolute best friend, so you know that just has to be done. So Raiders, what are we doing today? We have a live leafcutter ant colony. We're checking out some of their behaviors. We're then going to be looking for some tardigrades, and then also doing some ancient amber microscopy tonight. Welcome the heck in, everyone. I was also, before y'all rated in, giving a hard time for Jomio. Jomio, folks, is a big Manchester United fan, and he has very strong opinions on Scott McTominay and Anthony Martial. These are two players in the Manchester United team that Jomio has very strong thoughts about and does not like them whatsoever. Chad, do you know who scored this weekend for Manchester United? Scott McTominay and Anthony Martial. Yeah. This is a football with your actual feet, Smikes. This is football with your actual feet. You just get annoyed with it. You just call him the glass man. I'm sorry that you call him the glass man, Jomio. Hi, Claire. Welcome in. Not Roy Kent. Roy Kent is breathtaking. Big gaming. It's breathtaking. Real football, exactly, Quaff. Now, we have right here a leafcutter ant nest, and you can see there's the trash that they brought out. So the trash is dying and decaying fungus. They have been using and bringing out a lot more of it lately. And my nerves are about this. Gwen will tell you, I was nervous that something was going wrong with the colony. And a day or two later, I saw the queen's body in the nest. Dead. And then later that day, I wanted to go in and collect the body so we can preserve her. And then she was gone. I don't think she's alive. I think we lost the queen. I've already been put on a wait list to try to get another colony. When we got this one, there were two queens in there because these ants participate in polygyny. Hi, Hugin! Welcome in, Hugin. Polygyny, which is multiple queens. There were two. No, Claire, she was upside down. No. She was done. She was done. I'm not happy about it. I'm not happy about it. But we took it. So I'm thinking that there might be another queen still in there, but the likelihood is very low. Horizontal napping, Hugin. It's the best way to nap. I've also napped vertically before, Hugin, including today. Stone Monkey Research, the queen is dead, long live the queen. Unfortunately, to Stone Monkey Research, that's it for that colony. If the queen is in fact dead, there's no replacement for it. Freedom Dow, I'm researching colony operations for machine learning. Nice, Freedom Dow. That's really cool. There are some machine learning algorithms where you incorporate movement of the ants and figure out how they move and how that corresponds to their behavior. Yeah. Quaff, they don't kill their queen, do they? So it depends, Quaff, of what you do with them. By the way, folks, she didn't remind me, but the lovely Moo Hoodles is in his house. Also go try lovely, amazing Moo Hoodles. Moo Hoodles, folks. Another science communicator extraordinaire. We love you, Moo. We love you, Moo Hoodles. So, chat, what else did we identify in this particular setup? Oh, with the queen. So, Quaff, there are examples of killing queens on accident. Meaning, like, so I've seen some documentaries on leafcutter ants, and actually I know one of the gentlemen who worked as a consultant on these documentaries, and there's a whole scene where they're holding the leafcutter ant in their hand, the queen. And what's happening when you do that is your odorants get onto the body of the queen. Okay, and what that creates is the queen might not be recognized later by the colony. So that is really, really unfortunate because he told people, like, he told the producers, don't do that. And they're doing it, and they did a take, and he's like, okay, they're putting back the queen. And then all of a sudden, what did they do? What did they do? They did it again. The gentleman told me they did 6 takes. They did 6 takes of them holding the queen. Hi, Lilith Hobo! And constantly covering, like, the odorants there. And then what ended up happening? They shipped the whole colony to a site to do, like, a bunch of videography. And when the gentleman was unloading, you know, he unloaded everybody. Then all of a sudden he looked up at one of the tubes and he saw a body being moved. And that queen's body was being carried between the tubes. Because, why? They killed her. The workers killed her, Quoth the Raven. The reason that they did that was, again, she smelled no longer like a queen. She smelled like human. And that's how they tell individuals of the colony. Which is quite fascinating. That's how it works, right? Poor ants, let's say there's no second queen. How long do they have left? What's the procedure you will follow? So, Gwen, they will have... It depends. Gwen, I'd have to look inside and crack open the fungus. I don't want to do that, Gwen, because if I crack the fungus, the edges start to die of the fungus, so I don't want to do that. Instead, what I would do, like, because the reason I want to do it would even suggest it is because if there's any larvae in there, that would inform us how long the colony has. If there's no longer developmental stages, we're talking we're talking 3 to 6 months. If there are developmental stages, we could be talking 6 to 10 months. Championettes, thank you for the follow. Welcome the heck in. How you doing, Championette? Guys, also go check out the lovely, amazing Supernatural Writer who is in his house. How you doing, Supernatural Writer? Welcome on in. Gwen, if we get a new queen, we cannot add the new queen to the existing colony. They will eat her because they're 2 different colonies. So what we'd probably do is we'd have a second camera on this batch and we'd have the first camera on the other one. So that's what we'd end up doing, I think. Yeah. Human smell must be really gross to ants. So Lilith, that's why they attack you. When you if you were to stick your finger in an ant colony, the reason they start biting you is because you don't smell like ant. You smell like you, and you are invader. And so that's what happens. Why would you listen to professionals? So Quoth, actually, if you want, Quoth, we're rabbit holing, but if you want, I can actually show you the scene. Quoth, are you interested in seeing the scene where this happens? I can show you where they went wrong. There we go. They did go straight... Well, they didn't go actually French Revolution on this one, Hugin. They didn't end up... the ant did not get its head chopped off. Okay, here we go. That would be fascinating. Let's do it, Quoth. Whoa! I just started the pre-screen there. Boop. And, okay, here we go. There we go. So this, right here.
What in here?
So actually, this documentary, Quoth, is... I love this documentary. It's an hour and 28 minutes long. And we actually, Quoth, maybe a year and a half ago, Quoth. Maybe a year and a half ago, we watched this docu, maybe over the course of 3 streams. And the reason we did that was because we dissected the science behind it. Yes, I will post a link. Also, Freedom, if you would like, these are always posted in our Discord as well. There is a section on the Discord called Mentioned on Stream. And if you click on that, that little section, all the videos that we watch are all there. And then there's, right underneath it, there's a microscopy imaging section. And that imaging section is for all the pictures that we take on stream. Those are in that section. So if you're interested in it, there's a lot of cool stuff there. So here, Quoth, they identified the queen.
Yes,
There she is.
And the way they did was, incredibly amazing the way they identified where the queen was, how to find the queen, what to do with the queen. There was a whole bunch of stuff that they do. And it's an absolute amazing thing to see. So the gloves, so Quaff, yeah, the gloves prevent your smell from getting onto them. And it's that you want to use gloves when handling them. Honestly, though, you don't ever want to hold on to them either, because they're pretty fragile. But if you have to, you want to wear gloves. Hello, Optic Nerve. Hello, Optic Nerve. By the way, folks, if y'all like science, please also go follow my other amazing friendo. Her name is Neurolisha. She is live right now. She's almost at a follow goal. She's at 79 followers. She's looking for 100. She's a neuroscientist. Y'all can please go check her and drop her that follow. I think y'all will be very, very happy with the lovely Neurolisha. All right. Is that Carlo? Yes, Carlo is on the top right. Carlo is on the top right, folks.
With the queen. Excellent. 1 ant in 2 million, we found her.
Through tons of earth, we've managed to find the most important ant of all.
So the gentleman, Quaff, who had the gloves, is the expert that I know. So he's actually the world's leading expert, Quaff, on leafcutter ants. He has a shop called something like leafcutterantsforsale.com, and he's the only legal import-exporter in the world because he has licenses from Trinidad and Tobago. So he has a house there. He goes and collects full leafcutter ant colonies. He then brings them to his place in Scotland, raises them and sells them to zoos and research institutes. And he is an absolute riot. He's a Scottish gentleman.
It's a great relief to the whole team.
There he is.
And it means our project can go ahead.
So this is what we've been seeking in all our mining. This is
Alright, so here at Quaff, this is the unfortunate time.
the queen of the colony, and I'm going to very carefully pick her up. She's surrounded by attendant workers who are biting me now.
First of all, she's huge. So Quaff, look at the size of her. Like, that black-brown mass is the queen, relative to the size of his hand. Right? She's quite large. There are some little workers around her, but that's her right there. So the queen ants are quite beefy, as it were, right? So Quaff, here's the catch. When the small ants nibble, it's more of a tickle than anything. It's not anything that intense. When the queen at this stage, so this colony, Quaff, is about 1 million ants in size, is what they estimated. She's at that point no longer aggressive. Before she actually got mated, and like during her mating flight, she's still actually quite aggressive. But after mating, she's no longer aggressive, and she just, does her thing. And so that's one interesting facet of her behavior. So she won't be biting. It's just the nurses, which really aren't like that crazy of a response.
Now, ow. But she's in there. That's the egg-laying machine that's at the heart of this colony. And there she is, ready to go on her. We're going to stick her onto a really nice piece of fresh fungus. Very carefully, very gingerly, just plonk her on the top there.
So you can see Andrew's very upset by this. You can see how quickly he's trying to get this to be over with. So the behind-the-scenes Quaff that no one talks about, because this isn't really like in the video or anything at all. But what ends up happening is they take her back for like a recording session. So let's see. Let me show you what the apparatus actually ends up looking like. because the whole apparatus they have is quite remarkable. So they have tubes upon tubes upon tubes. So you see this entire background cloth with all these connections, like these glass tubes and boxes. Each of them have fungus, and they're simulating tunnels. And then on top there's a big dirt arena, and that big dirt arena is where they actually do foraging behavior. Here we go. This is a better shot of what they're set. So Quoth, this is like the coolest ant setup you're ever gonna see. Absolutely remarkable what they can do here. And they put RFID tags on the workers and the soldiers, and so to identify movement of the ants. It's quite remarkable. Zip on tubes. Hi, Hugh. How you doing, Hugh? Antropolis Astronomy Show. How are you doing, Astronomy Show? I saw one. It wasn't close. I saw a good one. It wasn't closest. Yeah, I've seen a few good ones too, Smikes, but this one is like next level. Just like our friend Astronomy Show is also next level. Hi, Canada. They put RFID tags on the workers, and they were actually able to track their movement in 3D space in all these boxes and where they were going. So, what ended up happening, Quoth, is that when they unloaded everyone, he saw one of these tubes, and the worker, there was a trash or sanitation cast carrying... Hi, Travel the World! Hi, Travel the World. Carrying the body of the queen to the trash pile. Now, the trash room, there are dedicated trash rooms in here. They are the lowest chamber. They are right here. That's the trash chamber. That is represented in the colony as well. When there is a leafcutter ant colony, the deepest chamber will have the trash in it. And that's because if there's any cave-in or collapse, imagine if you had your trash room high. If there was a cave-in, right, your trash would fall on your fungus, which you're supposed to be eating, or your babies, and everything would get infected and die. And so I can't tell you how they know this, but the ants somehow know to keep their trash chamber in the lowest possible chamber they're going to dig towards. And so it's just another amazing, Quoth, you and I can agree, yes, we don't want trash on our food, but the fact that that happens is quite remarkable. Hi, Tactics! I don't know if I've said hello to you, Tactics, but I love your face, Tactics, you amazing soul. Do they build new trash rooms when the other gets full? Yes. So, it turns out they actually will seal off the existing trash room, and then they will build another one down below. And so they'll have multiple ones. And apparently the trash, as it will eventually break down and get reabsorbed into the soil, and they can eventually open up the chambers as well. The ones that didn't sort of chance survival, presumably, Chris, but it was like a lot of ant species have trash chambers. It was just here in these particular ants, they are so specialized, there's a whole cast dedicated to them. It usually tends to be the older cast as well, Chris. So the younger ones do different tasks, and then when they're closer to their last limbs... Why are you angry, Andrew? Why are you angry, Andrew? What did I do, Andrew? What did I do, Andrew? What did I do, Andrew? As they're on their last legs is when they move over to being like the sanitation cast. And actually, some in the wild. Hi, Nona Silverstar. Welcome in, Nona. He lost at a game? Possibly. Wait, who lost? Dude, I'm sorry, Andrew. Andrew, let me know if you need anything. Let me know if you need anything. We should watch... We can, Gwen. We can do a deep dive in this one. It's a really, really cool video. And again, I got to talk to the gentleman. I met him, and he told me a bunch of stuff about it there that isn't in the video. And then I did a bunch of research as well on these, so I know a little bit extra on that front too, which I think is really cool. And colors almost seem to be like a 3D circuit boards. Yep, I agree, Can. Gato, that looks beautiful, by the way. Nona, how are you doing today? It is great to see you, Nona Silverstar chat. We met Nona Silverstar through the lovely and amazing Gato, aka the Stream Scribe. Guys, go check out the lovely and amazing Nona Silverstar. Antonio's very channel, which looked away and was like gargoyle. Quoth.
The spell is broken. We live again.
Quoth, here's the thing. Quoth. I have a secret project I'm planning, Quath. A secret project. And it's not so secret because I'm going to tell you what it is. I'm going to animate all the core gargoyles and have it be a redemption. I've already have the digital files. I need to draw a few more scenes. And then we'll have a whole series of the gargoyles, like, pop up as an animation, which I think would be really cool. I had fun today with Carlo, and then I got run errands with my dad. Hey, no, that sounds lovely, though. That sounds like a great day. Yeah, so Quath, that is a very long type of answer for you. Well, Big Game, I didn't tell you, like, all the extra secrets, right, about, like, how, you know, I have to make the stone versions of them and then have them pop out and animate the stone flying off. There's a lot I didn't tell you, Big Gamey, about it. I need to find the series. It's on Disney Plus. No, it is on Disney Plus. By the way, folks, we do also have a new animation you can trigger. "3D print!" Exclamation point, 3D print to trigger the 3D printer that prints Kevin. No, not Disney Poosh, but Gamey, Disney Plus. Big Gamey, I met someone who said Disney Plus non-ironically. Like, he thought it was actually Disney Plus instead of Disney Plus. And so now I'm just hooked, and I just say Disney Plus. Yeah. So Chris, the Queen died. We think. So that was the plan, Chris, but this colony this was dug up, and it's about a 5 or 6 year old colony that is much more stable and less likely to, like, have this kind of death versus the early queens. Our queen was about a year old, and that is it's still a tenuous point in their lives. Now, there were 2 queens in there, Chris. My suspicion is that both are gone. I did contact the supplier, so we'll try to get an additional one if we can, although obviously no guarantees. It's just dependent on if we can or can't. We always say Disney. Really? Cliff, are you kidding, Cliff? Civil War? No. This, Chris, I think it was just sometimes young queens just don't make it for one reason or another. Well, one queen. So no, these species have polygyny, which means they have multiple queens. Not the one on the screen. This is Ada cephalotes. The one we had was Ada texana. Texana has multiple queens, but here in Ada cephalotes, they only have 1 per colony. If you introduce a new queen, the queens won't necessarily try to kill each other, but the workers will try to kill the new queen. No kidding, that's how you pronounce plus in German. Cliff, I don't think this gentleman had European influences, but I like the idea of it, Cliff. I very much enjoyed that. Maybe that's why he was saying plus. Again, I don't think there was any European in him, like, from recent times, but you never know. Yep, some do, Chris. Some do. And again, the Ada Texana do, but I actually haven't seen the queen since maybe the first day we got them, or like the first month, because then the fungus grows and they just bury themselves in the fungus and you can't see it. If you cut the fungus, Chris, the ridges where you cut it, that's those sides of the fungus end up dying and receding. So that's why you wouldn't want to do that. And then also, there was another question about Canada asking about the trash room. Here, because it's a fixed number of chambers, they just clean the trash room. And so that's what will prevent them from wanting to build a new trash room. But you have to make sure to clean them reliably. Otherwise, the colony might just die out, which is less than ideal. But yeah, there's really cool. So like here's the top of the chamber, Quaff. They actually have little ladders that take them up. And so there's like a whole dirt area, and they give them leaves, and it's like the whole arena they built was amazing. Yes, Chris, I've already contacted the vendor that I got the original ones from, because we already have the permitting paperwork worked out. And so they said they don't have any right now, but we'll be added to the wait list. That's the plan, Chris. So fungus rooms, trash rooms, brood rooms, queen's chambers, others. So astronomy, in the most basic of ways, there are chambers with fungus, and there are trash rooms. In the most basic of breakdowns. Most retard, no worries, I love you, Carlo, the stream, subscribe to Carlo. So, astronomy show, even in the Queen's Chamber, the brood rooms, all have fungus in it. So, all of their rooms... Let's see if we can find... This is actually really cool. About just real quick, this is the RFID chip chat tagging. So they tag the soldiers and they watch where they went in 3D space. And that I thought was a really, really cool, really remarkable setup. So they figured out where the soldiers move in relation to the Queen as well. So the Queen's chambers, astronomy show, they are just wherever the workers decide. The workers will actually want to move the Queen around. If they don't move the Queen, if you prevent them from being able to move the Queen by a thing called the Queen extruder, so that is essentially you shrink the doorway of one of the tunnels so that the workers can get in and out, but the Queen cannot. If the workers decide that they want to move her, but you have a Queen extruder on, what they do is they chop her up and move her that way. And because the workers can't realize that they can't put her back together. So they chop her up and move her body parts to the chamber where they want to go. But then that's kind of it, right? There's nothing else that could be done. So yeah, it's a little cringy, unfortunately. Now then, the chambers. Let's find one of these just fungus boxes that they open up. Here we go. So these are what the chambers look like, astronomy show. The core is then probably where the Queen is. Hi, Bex! Welcome in, Bex. How you doing, Bex? So astronomy, all the boxes, if you look inside, look like this, except for the trash room. So you see how it's this almost, it's not really a honeycomb-like shape, but it's like a spongy shape with all these little holes. All those holes, they can climb into. And so it's a tunnel system in the fungus. And so that's where the brood are kept. The Queen is somewhere in there. There's workers in there. There's soldiers in there. There's workers on the outside building the fungus. There's workers on the outside getting rid of the dead fungus. It's a city in a city, almost. Because I would argue that the chamber itself and the tunnel system is part of a city. But then inside that chamber, there's a second city, which is the fungus. Nona, I try hard, Nona. I try really hard. And then look. Whoa! Whoa! Oh my God. Whoa! We get silly here, Nona. We get very silly here. So that is, I think, one really remarkable feature of this system that they have. Again, not usually recognized of how it works, but still really cool.
And I can see some new ones too.
Beautiful architecture. And Chris asked if they do tours. So Chris, this was in England. Hi, Mixeneth! How you doing, Mixeneth? Welcome in. Chris, this took place in England. Ooh, what year was this? I think this documentary was 2013. I can't remember, Chris, if this was utilized in any different way. Like, I don't know if they kept it after the documentary. Although it would have been great if they... I don't know if they have tours of it or not. Knowing that it would be death to the Queen, why would someone do it? Holding the Queen coffee? Because they want to get a good shot. So, Chris, in theory, it could still be going. Because leafcutter ant queens, if healthy, live 30 years. Again, there's a high rate of mortality at the start of life. during the mating flight, after the mating flight, up to the first 3 years of life, they have a higher mortality rate, the queens do. Usually it's because of having a stable colony, and then sometimes they're just not strong enough to survive and keep laying. There's a bunch of weird things. Yeah, like humans or other animals too, right? But once they make it over that initial hurdle, then they'll make it, then they're pretty set up. So that, the max that we've recorded is about 30 years, but you're talking anywhere like 20 years easy. So in theory, the colony should still be going if they decided that they wanted, to keep it going. That's the big thing. But yeah, it's quite remarkable. Yeah, coffee, I think it was just they wanted a good shot. Here's the trash room as well, for y'all interested. Moist Halden, welcome the heck in. First time chatter. Moist Halden, folks, won a pin today over on our friends Moko Made stream, a SciAnts pin. Chat, the Moko Locos are our stream team leaders. They are breathtaking, amazing souls, and they are doing a bunch of raffles for all kinds of things from the community. Moist, I don't remember which one Moist won, but we had all of our hard enamel pins for sale, including some of these. They were raffled off as well. They were raffled off, and Moist won one of them. Moist, welcome the heck in. We do science content here. Today is Microscopy Monday, and we'll be looking at tiny worlds under a microscope. In particular, we'll be trying to find some tardigrades today. Tardigrades are the animal that can survive the vacuum of space, which is quite remarkable, and just a bunch of stuff. We're also looking for we have Cretaceous amber today that we're going to be looking for as well through, and so there should be some insects in those Cretaceous amber. So that's some of the things that we're going to be working on today. I'm trying to see. So if you have any questions, please let us know. There was a question in the chat about the ant trash room. Can of DNA was asking about that. So this is the trash room inside of this particular colony. So you see there's actually a bunch of different sizes in here. So there's not one dedicated type of cast that look functionally different. They're just old members of the other casts, and they're relegated to being a trash cast. The reason for that is because they're old, they probably have a short lifespan, and that's why they're in there. Moist, that bread of yours is a national treasure. There we go. Thank you, Smikes.
This speeds up the breakdown of potentially harmful substances. But the ants aren't just dumping their garbage down here, they're also disposing of dead bodies. Over on...
My face is a tardigrade? Saw an article where they're going to splice some tardigrade DNA with humans. So, Moist, so tardigrades can survive very extreme, like environmental factors. And so one thing that researchers are trying to do is figure out what is the genetic basis of how that works, right? And so it would, like, trying to figure out, like, maybe in human cell lines, that's maybe how it works, is they're trying to move over human cell lines to respond to certain genes. The ants made their queens similar to... So we don't know canned DNA. There is one tardigrade from Astronomy Show, and there's a second one here as well. So, Cam, we do not know how the casts are developed yet in ants at all. There is a suspicion. So the reason that's weird, Can, is because in bees are put into a little chamber, and in their chamber they're fed different diets, and those diets are constrained to that animal. So if there's someone eating royal jelly to make them a queen, that's all that's there. In the ant system, they're not in these little units. They're just with a bunch of other individuals as well. And because of that, there's no way to actually regulate who gets what food. from a like given food source. So what we think might be happening is the nurses that are trophallaxing or regurgitating food into the larvae, they're spitting epigenetic modifiers into the mouth of the larva to create a particular cast. And they can sense what cast to make based on densities of certain types of workers, whether that be workers, soldiers, cutters, queens, or what have you. And for most ants, there's a seasonal pulse of queens or males, and so there's also seasonal cues coming in, unlike with the bees. So we're not entirely sure yet how that works, which is disappointing. You would have thought we would have known it by now.
On this side is the graveyard. Now, this is actually very interesting. The ants, of course, don't live forever.
The catalyst generated by the nurse is pretty... Exactly, Chris. So, like, the soldiers come the latest because they're the biggest and they require the most amount of nutrients. So it's like there is a thresholding based on what you have available in the colony as well. Let me spell it for you, Quoth. There you go. They can roughly count, yeah. Make some different shape sizes, even though, so it's all epigenetics that modify. So the DNA, like, expression has changed, so the accessibility of genes within the DNA strand is changed versus there being, like, differences in the actual genome. The DNA sequence is the same between all the castes and worker caste, queen, what have you, but they are still come out very different due to the epigenetics of it.
And when they die, the remains are taken down and dumped out of the colony. And that, in my hand, is just the dead remains of...
And what we've already done this before, but I think next week we'll do it. We'll take a look at the trash pile from our colony and see what's in their trash pile. Would that imply that nurses are likely super aware? Yes. I think I would echo that sentiment, Chris. That actually, if you look at their brains, if you look at the brains of leafcutter ants, so here, for example, is a 3D printed model of one of our leafcutter ant brains. What you can do is you can take images to measure volume of the brain. You can then also measure... Gwen's got it. Thank you, Gwen. You can take volumetric measurements of particular structures of the ant brain, and then say, like, you can calculate what percentage of the entire brain is that one structure. And so I did that, and what you find, Chris, is that the nurses have the highest percentage of mushroom body relative to the rest of their brain. Now, their mushroom body is their learning and memory center, which would be the one also for, like it's hypothesized to scale with higher social interaction. So ants inherently have bigger mushroom bodies than fruit flies because they're more social, but those nurses have the biggest ones, presumably because they're the most social within the colony. And presumably it makes sense, like from what we're saying, that they would have that largest brain structure because they need to be able to perceive those threats. Epigenetics also... So the brains themselves aren't bigger, Canada. It's the proportionate breakdown. So, like, their mushroom bodies are bigger with relative to the rest of the size of their brain if you compare it to, let's say, a soldier. But that is also... But that size relative change is also epigenetic. Yes. What organisms live in the trash pile? So there are, astronomy show... bacteria that break down the trash and like what's existing in like the actual fungus. There are midges and springtails, and those actually help break down the decaying plant matter. So those are actually beneficial for the colony to have inside of like their trash chambers. If there are mites, it gets really bad. Mites, there's two kinds of mites, astronomy show. There's food mites and parasitic mites. Parasitic mites are, as the name implies, parasites that can kill and take over the colony, and they will very much so nerf the colony if you have mites. Food mites tend to prefer fresh food and not rotting fungus, although there are a few that do. But likely if you have the food mites, they'll also start eating the fungus, the living fungus, and then the society of the ants break down. So you have to be very, very cautious when picking and examining the individuals within the colony, and like how clean you're keeping your colony, because otherwise you could get invaders in there. There shouldn't be beetles or any other critters in there really alive beyond the mites. And
Literally hundreds and hundreds of ants of all castes. There's small workers, large workers, soldiers. And so when the ants have no longer any function, and when they die, they're simply taken out and dumped.
And they are, the way the ants know that an ant is dead is through smell. There is an odorant called oleic acid that is released from a dead ant, and that's how workers know to take it out to the trash pile.
The main reason why ants have to keep the waste and the dead remains of ants out of the way of the colony is that when you're in such high abundances in the colony, you don't want any diseases to spread, and so you have to maintain your environment. It has to be clean, so anything that could possibly rot is removed.
Seeing the dump and the graveyard really brings it home to me just how sophisticated the ant colony is. It's easy to see why people have looked at the ants and thought they were seeing our own world reflected back.
I've heard if you put that acid on a living ant, they will take it. They will. Yep, they will. They will pick up the living ant, move it over to the trash pile, even though it's still alive. Eventually that odorant dissipates, and so then they're allowed back into the colony because it's a volatile. So it means it evaporates off, and eventually, if it evaporates off totally, then they're okay to be let back in.
We've even used the language of our own social structures to describe ant society. Workers, soldiers, the queen. But is ant society really organized in this kind of hierarchy?
We'll examine the rest of this video, don't worry, chat. And doesn't the living ant just sort of lay there because she thinks it's dead too? It depends, McZenith. It seems like that is species and ant specific. So there are certain castes and there are certain species that are more responsive to it than others. Hi, guest 2563, first time chatter. How are you doing today? Don't they put that on human food? I am not sure if they put it on human food, guest. It's what the ants have a much more concentrated version of it. But actually, no, guest, you're right. There are some cereals that have that. I think, for some reason, it's reminding me that, like, Trix has oleic acid. And so I think there actually has been done the experiment that if you give them Trix, they will stay away from it, or they'll move it into their trash pile. Which is kind of funny to have that happen. But yeah, there are some instances where they have that. Unlucky charms for the ants. Smikes, for the ants, they are unlucky charms. Alright, chat. Quoth, does that answer your question in like a very roundabout way? Quoth the Raven Nevermore. Chat, also go follow our amazing friendo, Irish John Games. Please, everyone, go check out our lovely, amazing friend, Jern. Game dev extraordinaire is Jern. Go check him out. You will never, ever, ever be disappointed with an Irish John stream. If, Quoth, that answers your question, let's go ahead and take a look at our aquatic samples and see if we have any tardigrades that we found. Wait, what do you mean that they were? Which one? What do you mean, Quoth? What do you mean, Quoth? Is a mono unsaturated fat. Actually, a lot of foods we eat, including nuts, olive oil, meat, cheese, and some veggie oils. Oh, gotcha. Thank you, Mixeneth. Plants produce the acid to keep the insects away. There are some guests that do that. There are also some actually that want the ants coming towards them, because the ants help farm their the plants as well. Like peonies, the flowers, they don't open unless ants, like these little tiny like sugar ants, bite the perimeter of the flower, which is quite fascinating, I think. Oh, gotcha, Quath. Gotcha. Quath, that was like, is Quath the Raven being self-deprecating? I was like, that doesn't sound like Quath the Raven. I was like, is that a near nerfable offense? It was not, Quath. We were safe. We were safe, Quath. I came to the realization that you were just being a silly willy and referencing something earlier. Thank you, Quoth. I get nervous, Quoth. But yeah, Chad, if y'all are interested in this, we could dissect the science behind this documentary. What we've done before is we watch the whole thing, but as we watch it, we pause it and commentate on the different pieces and the science behind it. If that's something y'all are interested in, we could definitely do that sometime soon, because it's quite a fun thing to do, I think. So we can 100% do that. Alrighty, folks, let's go ahead and check out our microscope. We'll be looking for some tardigrades to start out the stream before we start looking for our ancient amber. So we do have Cretaceous amber today. The Cretaceous amber is, again, 90 million years old. We have two more samples with Cretaceous amber that we can look at. We also have our Dominican amber, which is 30 million years old. All of which have some flavor of insect that we'll be looking at. Insect, arachnid, some kind of tiny critter. So let's go ahead and switch on over to the other camera. There we go. Now, what have I done already today, folks, with the samples? So, whenever the chair is like stuck for a moment, I'm like, what's in the wheels? I'm like, is it one of Baby Alona's blankets? It is not, because usually she has her pillow on this chair with blankets and like a washcloth and like all that fun stuff. She's such a silly goose. She was such a good baby today. She was such a cool baby. What we're gonna do is we're gonna put on a black background, so it'll be more visible to identify the tardigrades. There we go. We'll wipe it off.
The spell is broken, and we live again.
Got the wipeouts. Now, there is no guarantee that we will find a tardigrade. So, the best way and easiest way to find these little creatures, basically what you want to do is to take mosses and lichens growing on the side of trees that are dehydrated and put them into some water and let them soak for a while. If you do that, you might end up finding a tardigrade. So that is what I had done a couple of hours ago. I put some of these samples into some water. I actually used some pond water because it's already like at a pH and like an acid concentration that will prevent any kind of breakdown between these animals. Like they won't explode from different hydraulic pressure. And they've just, the piece of wood have been soaking. Now, they're not frozen, yeah, Quoth. Luckily, the pond hasn't frozen, but because we did have a freeze, I can't imagine that we would find a lot of creatures in the pond water right now. Last week, we looked, and we couldn't really find anybody. That's, again, not to say that there isn't anyone in there. There are presumably microorganisms, but density is an issue. Hello, Kodali.
Hello there.
Kodali, how are you doing today? We're about to start off with looking for some tardigrades. Again, no guarantee. What we're going to do is just continue looking for them across the next couple of weeks. Hi, Pragmatic Entropy. It is Microscopy Night, Pragmatic. We'll be doing some amber in a little bit as well. We got a hold of some Cretaceous amber, Pragmatic Entropy. So that's 90 million years old. We looked at 2 samples last week of Cretaceous amber, and we got another 2 samples to look at tonight if you're interested. But now we're doing a little bit of tardigrade hunting as well. Whether or not we'll catch any, you never know. We've gotten lucky before, and then we've also gotten unlucky. Now, we're going to try to see if there's any critters in here. The big catch, again, if there's none that we identify, what we can do is soak additional pieces of mosses. No, Pragmatic. Just 90 million. So 90 million. I spell it out because I know my words are always difficult to hear. You know, sometimes the southern accent jumps on in. Yeah, I think it's really cool because they're still, you know, from the era of the dinosaurs, right? Okay. Big Zenith, thank you for understanding. So, well, we do have some microorganisms here. I'm going to adjust the lighting and whatnot in a second. Not a tardigrade. But, chat, we have an amazing organism here. This is one of Smikes' new favorite creatures. And so this is why it was near intentional. Sorry, Oz, I'm just trying to lower the lighting. Here we go. That right there. They are moving very quickly. Let's... Come on now. You all see those little wigglings? It's not a planarian, Smikes. These are nematodes. These are like Ellie elegans. That SciComm character. These are nematodes. Right here. So they are very, very tiny. We're talking like...
The spell is broken, and we live again.
So the way that we know for these tubes, Smikes, these tubes are pointy at both ends. The planarians are not as needle-like in structure at their ends. So that's the way we can tell planarian versus nematode. They're also, Smikes, their reproductive organs are visible. So they're very see-through. V, hi V, don't lie, V.
Ah, you are wholesome as ferret. Yay!
And McZenith was right. Hi, Risto! Risto, don't lie to me, Risto. Chat, go check out the amazing Risto, who is actually breathtaking, kind, and amazing, and loving, and so handsome, and is soon coming out with a calendar, folks. A calendar to teach people about a nearby orphanage that he's raising money to help save. And he'll be shirtless in all the pages, folks. Here we go. That's a much better lighting right there. We'll even grab some pictures as well. But there they are. Those are our nematodes right there. And again, we're not getting the perfect visual on them right now because we are pushing the upper ends of the camera. But they are see-through animals. And what makes that really, really cool is because they're see-through, that's one of the reasons why they were originally studied, is that you can see the inside of the reproductive organs. And yes, there is an enamel pin of Ellie elegans. There's even exclamation point shock will bring up Ellie elegans on the page, which is always fun. It's a monthly calendar, Pragmatic. It's a monthly.
Victory screech!
Why we did... You got it, Smikes. Why McZenith, she's getting shocked, is because there was a paper that came out a few weeks ago that showed nematodes actually utilize electric fields to find their prey. They even use electric fields to jump out of water and parasitize adult fruit flies. Resto! I love you, Resto! Which is quite remarkable that they have such, like, a way for it. Similar to how sharks... They haven't identified the organ McZenith, unlike with unlike with sharks, where you know exactly, like, with that frontal nose organ that actually does the guidance of the electric fields. But they found that if there's electricity and moisture in the air, they're able to throw themselves out of the water at a particular prey target, without which, so if they don't have that prey target, what actually ends up happening is then, or with the electric fields, they can't jump out. So they need the electric fields to be able to do that kind of jump. Hi, Skip, aka Skip the Bell. Skip, are you being naughty today? Are you being naughty today, Skip? Chat, lovely Skip is in his house. Folks, go check out the lovely and amazing Skip, aka Skip024. Amazing father, gamer extraordinaire. Smikes, could you put that into our channel for the ideas so that I don't forget? But autopsies on nematodes are much easier, Pragmatic, I would argue, because we have the genetic tools available to test all the different cell types and what they do. Right? Because we have the genetic tools, we can go in and turn off any of these cell types and figure out what differences are yielded as a result of changing those cell types, and like whether they're being on and off. Now, that's what I think makes them easier. They're also easy to grow in the lab. But I get what you're saying as well, Pragmatic. Hi, RPG Fandle. How you feeling, RPG? Welcome the heck in. No, UV. Don't you say that to me, V. How are you doing, RPG? The readiness of those animals is much more limited in large animals. Or availability, I suppose. Depends. Yeah, it depends, though, Pragmatic. Because some of these are pretty... Again, if it's nematodes, we have a fair bit of information already in them. And identified, and so it's much, much easier to get those kinds of long-term, big questions about them. And you can see they're actively moving stuff out of the way. And so that's why we keep having to reset our bright field. They are very, very active and very difficult to catch an image of. Gaining more strength every day? Glad to hear it, RPG fan. Longer history with more complex life forms is what I'm saying, I guess. Ah, gotcha, Pragmatic. Totally fair. Totally fair. Yeah, these have been... nematodes have been studied since... I believe the 1930s. So one line of nematodes was isolated. So the wild type that you have is the exact same wild type from the 30s as it is today because they're clonal. So they're hermaphroditic. They self-fertilize. And because of that, it's the same strain that everyone uses. There will occasionally be males in times of stress to increase genetic diversity. But if there's no times of stress, then there's no need for males. Which is quite a fascinating piece of biology on its own. Once removed from the pond, how long will nematodes live until the food runs out? So astronomy, actually that's hard to answer because nematodes have this really amazing feature called the Dower stage. They essentially enter a state of stasis or hibernation. And they can stay like that for months. Like, if you put them on a dried out piece of agar, which is what they would eat, like dried out like a potato chip, like they can't eat any of it. You dry it out, they will shrivel down and live on it for months, if up to even a year. And afterward, if you rehydrated them, they come right back to life. So, I don't know exactly, like of this pond species dynamism, but for a fair bit of time, they can live. It's quite cool. The fact that we see biology eliminate us males in nature lets you know about humans. I mean, you never know, Pragmatic. They just don't need it. But Pragmatic, when they need genetic diversity, they bring back males. So it's just dependent on whatever external stimuli are present that they feel like they require. Like tardigrades but shorter. Yep. And again, you can identify them based on their extremely pointy heads and tails. The planarians that we've looked at in the past have much more like of a flat-shaped head and not as pointy as what these ladies look like. But quite cool. And actually, we would be able to sex them. The males are about 1/3 to 1/4 of the size of these hermaphroditic ones. So they are sexable given the size that we're seeing Again, they're not age matched. There could be age differences, but I don't see anything that looks like readily like a male. And again, they would be very, very puny on that front. So those are some nematodes for us. So they have both the ovaries and the testes, Smikes, and they have these really long reproductive tubes. That's a huge part of their bodies. So Smikes, on the Ellie animation, here, that internal coloring and stuff in Ellie is the reproductive tract, the yellow one. So it takes up a huge part of their body. So it's both the ovaries and the testes, because it's a different structure than in the males only. Because what you have in the females is a giant tube that goes like this, and they connect at the site of fertilization. And so they're just structured differently, versus the males just have a much thinner tube because they don't need to mix the eggs and the ovaries. So it's because of both in this case, Mikes. Again, just because the two structures are not the same between male and hermaphrodite on the male organ side of things, which is inherently also just like very fascinating, can I just say, of like from a structural perspective on these animals. But yeah, so we have some nematodes. Let's continue our hunt, chat, for some planarians. Let's look, not planarians, for some tardigrades. Let's see. No, actually, we can look at some more. These nematodes here, they were able to get them a little bit in better focus. There we go. If they just were to stop moving and swimming around so much, then we'd be able to capture one of these images, and you'd be able to see they have very, very pointy structures on their tails and their heads. And that's really the big delineator of how you can identify that they're nematodes. Interesting enough that they are on, they are just hanging out on these branches. They must have been in the dower stage. And that we ended up bringing them back. Relative to, yeah, so they feed Astronomy Show with this whipping-like behavior, and they can whip food towards their mouths. If the food is, alive. Usually in the labs, they're just fed like microorganisms, like, if there's any kind of small bacterium in the lake. In the lab, we can just give them E. coli. And interestingly enough, Astronomy Show, you can give them E. coli in the lab that's genetically modified. And when they eat the genetically modified E. coli, that can change their genetic information as well. In particular, that's how you can do genetic knockdown on them. So if you want to turn down a gene, what you can do is actually feed them a component of like a little bit of E. coli that then has a certain gene turned down, and when they eat it, they'll also have that gene turned down as well, which I think is really cool. So that's how they originally did all the genetic perturbations on these animals. Now you can do much more fancy things. You can CRISPR it. So, because the thing is, if you're feeding them, you're not getting tissue specificity. You're affecting all tissue equally, and maybe you only want to affect the brain or the the reproductive tract or something else, which is very difficult to do if, you're feeding them the component, but pretty remarkable nonetheless, right? I don't know, they're also just kind of fun to watch, I think. There's just such interesting locomotor movement they have. They'll also use their tails to latch on to some of these substrates Astronomy Show, which is, I think, really neat as well. Big fan of rotifers. So, Pragmatic, we have caught rotifers in the past. I don't imagine we'll catch any today. We will be doing more pond water as the weeks come by, but Pragmatic, we had a really bad freeze Saturday night to Sunday morning. So I decided not to grab any pond water today. Instead, we just went for these pieces of lichen and pieces of tree debris, like the tree bark, and trying then to see if we can find a tardigrade. Yes, yes. A month from now, I would hope that we do. We've had mossy wort plants as well, pragmatic, that they are full of these little capsules that capture little insects that they then digest and eat. And it was actually fun. We dissected open some one stream and let out the little critters. It meant that plant wasn't getting its meal, but you know, it was still fun to do. Alright, so I know y'all can't see right now as well. But because I'm searching around and seeing.
You are
Mon! No.
as ferret.
Don't you dare.
Yay!
Don't you dare, Mon. Chat, if you're not checking out Everyday Gamer M, aka Mon, aka also Monique, is a breathtaking game dev, artist, and gamer. Please go check out the amazing Mon. Also, one of our most favorite Irish streamers in the world. Not just on the internet, but the entire world. Mon, how the heck are you doing? Mon, lovely stream today. As always, Mon, we are on the hunt for some tardigrades right now. We did find a bunch of nematodes that were here. They are, so they're swimming around. They are hermaphroditic animals, which is inherently really, really cool, that they self-fertilize their embryos. No, you have not, Gwendolyn P. Gwen, yes, Gwen, we did all the amber before.
It's alive! I've always wanted to say that.
That's absolutely, no, Gwen, I'm kidding. Gwen, we're still hunting for the tardigrade, Gwen. Don't you worry, Gwendolyn P. Don't you worry, Gwendolyn P. We love you, Gwendolyn P. I'm researching some crochet because I'm making Alona a doll needs to have a lab coat. Yo, Mon, that'll be so cool. Mon, how was your rest of your day? I hope your day went well. Again, I'm just currently searching for some tardigrades. Going between sample wells, trying to see if we can find anything. The way that we do this, Mon, is fairly straightforward. Like, anyone can do this as long as you have some flavor of microscope. You get some tree lichens and mosses, and rehydrate them. So just put them in some water and let them soak for a while. And if there's any tardigrades, they'll just swim on out and they'll be exploring. Really good, thanks. We went for dinner. After stream. I was meant to have an early night, but I'm up writing. What are you writing, Mon, if I may ask, Mon? I bet it's cool stuff, Mon. So, this is Gucci. Hi, Jern! Chat, you know him. You love him. Irish John Games is in the house. Folks, if you're not already checking out the video game Rise of Piracy, you're not doing life right, folks, make sure to wishlist Rise of Piracy, available now on the Steam store.
Ahoy there, Beckon scientists!
Hit the wish list! Ideas for the next game? Oh, Mon, I'm excited, Mon. Chat, Mon, did you know this music is played by Irish John Games in his bathroom? Mon, did you know this, Mon, because you're also in Ireland. Did you know that Irish restrooms are the most acoustically perfect restrooms there ever are? John actually got onto this because, Mon, in Ireland, the bathrooms are circular. The toilet is in the center of the bathroom. And John stood up in his toilet like this. And played that music. And then Mon, in this follow-up video, trailer for Rise of Piracy, Jern also sings. Wait for it, Mon. coming, Mon. It is glorious, Yulo. How you doing, Yulo? Right now. That's Jern right there. Chat, go follow him. The Fancy Pants Pinky Out shout out is right now, chat. Voice of an Irish angel indeed. John, how the heck was the rest of your stream? Thank you for doing everything you do. You are one of my most favorite people in the world. I love your face, Irish. John, sing your melodic screaming. Pragmatic. With Jern, it's always the voice of an angel. The voice of singing and beauty. Jern, we're doing alright. Jern, we got a new animation live today.
SciAnts know you. You're the cutest, and that you deserve it, Juno. You a star over here. I will reverse the card like Uno.
You're breathtaking.
Bill, yeah, yes. Jern, exclamation point 3D. We now have a 3D printing animation where Antonia 3D prints Kevin. Andrew, I now know what that means. It's pretty cool that Gucci. I know. Gucci. Mon, you are Gucci. Not Jern. Jern is so... Guys, that Irish John. Look at Jern. Look at Jern. Look at Jern. Oh. You triggered it, John! Nice job, Jern. Nice job, Jern. Did it again. Triggered it. Gwen, I had no idea what a Juno was. I thought that was a movie. Right? I thought it was a reference to the movie. John, nice job. Did you animation waves? Mon, what was the newest one? The Zelda was pretty cool. The Zelda turned out alright. Oh, and we saw the Easter stuff.
5 Kevins.
Gucci.
There's Jern as Kevin. We do Mon. We kept the Easter stuff until this coming Sunday for Orthodox Easter. So there's still all the Easter stuff too. There's always
Where's
fun.
my drink?
Jern, we're doing alright though. We're doing some Microscopy Monday. We're checking out this pond water, and we're hunting for not just eggs, but tardigrades. We'll see if we find any today. I didn't know what we would find, to be totally honest, because it's still on the colder side, but we found some nematodes. These are C. elegans. They have both male and female reproductive organs. So they can self-fertilize, which is quite remarkable. Then we're going to be looking at some amber Jern, and then tonight we're going to be going over to Nerduino. Nerduino is going to have me on to chat SciAnts and SciAnts today. You want to keep Kevin all year round, Gwen, for the egg? But Gwen, for Thanksgiving you have a turkey. Gwen. Now you trigger both and he's cooking. You trigger both and he's cooking, right, Gwen? I guess he could throw chocolate at your face for Valentine's and maybe like peppermint or other things. That is at 11 p.m. Eastern astronomy show with Nerduino. 3D, yes. Nice job, Jern. Do you play with light and polarization in your microscope streams? We do, Pragmatic. All the polarity stuff is on the other microscope. Gucci. So it's on the light microscope. That one has a different polarization filters. This one doesn't have any particular filters, although we could purchase some, but we don't have any currently. Yes, Mon. Hank works on Nerduino streams later. You have to ask Nerduino, John. Because I'm not in control there. If there is a redemption like that, then you'd have to ask Nerduino. You have to ask Nerduino. Can we get a double Hank? John, I am 100% for it. You just have to ask, you have to ask Nerd. Jaren, we're doing alright. Jaren, we got a new animation live today.
SciAnts know you. You're the cutest. Tell them that you deserve it, Gino. You a star over here. Oh, reverse the card like Uno.
You're breathtaking.
Bill, yeah, yes. Jaren, exclamation point 3D. We now have a 3D printing animation where Antonia 3D prints Kevin. Andrew, I now know what that means. It's pretty cool that Gucci. I know. Gucci. Mon, you are Gucci. Not Jern. Jern is so... Guys, that Irish John. Look at Jern. Look at Jern. Look at Jern. Oh. You triggered it, John! Nice job, Jern. Nice job, Jern. Did it again. Triggered it. Gwen, I had no idea what a Juno was. I thought that was a movie. Right? I thought it was a reference to the movie. John, nice job. Did you animation waves? Mon. What was the newest one? The Zelda was pretty cool. The Zelda turned out alright. Oh, and we saw the Easter stuff.
5 Kevins.
Gucci.
There's Jern as Kevin. We do Mon. We kept the Easter stuff until this coming Sunday for Orthodox Easter. So there's still all the Easter stuff too. There's always
Where's
fun.
my drink?
Jern, we're doing alright though. We're doing some Microscopy Monday. We're checking out this pond water, and we're hunting for not just eggs, but tardigrades. We'll see if we find any today. I didn't know what we would find, to be totally honest, because it's still on the colder side, but we found some nematodes. These are C. elegans. They have both male and female reproductive organs. So they can self-fertilize, which is quite remarkable. Then we're going to be looking at some amber Jern, and then tonight we're going to be going over to Nerduino. Nerduino is going to have me on to chat SciAnts and SciAnts today. You want to keep Kevin all year round, Gwen, for the egg? But Gwen, for Thanksgiving you have a turkey. Gwen. Now you trigger both and he's cooking. You trigger both and he's cooking, right, Gwen? I guess he could throw chocolate at your face for Valentine's and maybe like peppermint or other things. That is at 11 p.m. Eastern astronomy show with Nerduino. 3D, yes. Nice job, Jern. Do you play with light and polarization in your microscope streams? We do, Pragmatic. All the polarity stuff is on the other microscope. Gucci. So it's on the light microscope. That one has a different polarization filters. This one doesn't have any particular filters, although we could purchase some, but we don't have any currently. Yes, Mon. Hank works on Nerduino streams later. You have to ask Nerduino, John, because I'm not in control there. If there is a redemption like that, then you'd have to ask Nerduino. You have to ask Nerduino. Can we get a double Hank? John, I am 100% for it. You just have to ask. You have to ask Nerduino, John, because I don't want to speak on behalf of Nerduino. Are the nematode species from the bark or the pond water that hydrated the bark? So Twitch Addict, great question. The pond water, I don't think it's from the pond water. because I just scooped from the top of the water.
Gucci and the Hoochie Mama.
Hey, Gucci and the Hoochie Mama. Because I pulled from the top of the water. We've done that in the past, and there haven't been these many nematodes. And so my assumption is that they are from the bark, which is also fascinating because we've put bark before, Twitch Addict, and never seen this many nematodes. So there's something funky with the bark that we used, or so, I don't know, but it's Gucci. I don't think it's from the water. Nematode, yes, John. I'm so sorry.
Victory screech!
Is that okay, John? The
Gucci.
Gucci.
There is a John. One of our mods did one that if it's popped up a few times, but I think if you hear that one, you will have a huge chuckle.
Gucci.
That was also done by one of our mods, Bit Gamey. But not that one. So is this like a swimming pond or like a swampy water? Bex, you should not swim in this water. You should not swim in this water. Whom? You, Smikes. There's the one with the beep is so funny to me.
Gucci.
Not that one. Eventually we'll hit that one again. Every time, if you want one of the Gucci's, they don't come up.
Gucci.
Played my Gucci sound for her friend. She got so triggered because she's convinced I modded my voice. Mon, you did not mod your voice. You have a beautiful voice.
Gucci.
I will fight them, Mon, to suggest something like that. That Mon's voice was modded. How dare. So yeah, there are nematodes. I'm going to zoom back out. We're going to continue on and move around and see if we can find any more. Although this is a good shot, too. Again, all the pictures will be on the Discord later on if you want to look through and try to find any samples. Like extra, I'll just put them onto... There's a whole section on the Discord that's called Microscopy Images.
It's all Gucci.
And that will be for you to... you can check all these things out. I will admit, I speak a little different on Mike's stream, so I'll give her credit to do that. I mean, Mon, we all do, you know, because we gotta play it up a little bit, you know. We gotta be excited, Mon. We gotta be fired up, Mon. We gotta be, you know, as John says, you gotta be Gucci and the Hoochie Mama. Which Irish John has been informed of the inappropriate nature of that phrase. Can't believe he said that. Can't believe he said that, folks. Send that to a family-friendly stream. How dare. How dare, Jern. How dare. So what I'm just doing, I'm moving around the debris and seeing if we have any tardigrades in here. And they would just be identified by shape and movement. Again, the nematodes are very full. And Twitch Addict had the question of, are the nematodes from the water or from the tree bark? So Twitch Addict, I used a couple of different kinds of tree bark.
We'll all be Gucci.
And because I used a couple of different kinds of tree bark, that should be our answer for it. Billboard Supermodel is here. Your girl isn't. Thank you for letting me know. Gwendolyn P. Chat. A Billboard Supermodel isn't in his house. Lovely and amazing. Yogurt Garrel, the most breathtaking supermodel across the country of Canada. Folks, she's on billboards from Saskatchewan to Quebec to Montreal and so much more. Chat, if you're not already checking out the amazing Yogurt Garrel, please go drop her that follow right now. She is also a Tetris 99 professional.
Curious and curious.
First place winner extraordinaire, one of our best friendos. Yogurt Garrett, how are you doing today, man? Welcome the heck in. I hope you're feeling better, Yogurt. I hope you're feeling better. And if anyone hurts you, we will destroy them, Yogurt Garrett. We'll destroy them. Yes, indeed, Yogurt Garrett. How you doing, Yogurt? Let's see. Hold on. We had something here. My son's friend in high school went swimming in the creek, which is common practice, ended up almost blind from an amoeba getting his eye. Yep. Twitch ad. You know what else makes that worse? If you wear contact lenses. If you wear contacts and you don't clean out your eyes and you don't remove the contacts, in between the contact and your eye is where those creatures will live, and that's how you can lose an eye. So be extremely careful when wearing a contact. He did. Yep, that's probably part of it. So Twitch Addict. I'm trying to find and get it in view, but this is another one of another well. And you see how in this one, Twitch Addict, there's no nematodes.
It's all Gucci.
There's no nematodes swimming around. It's the same water. It's just a different type of tree bark. And we don't see any nematodes. So the nematodes are coming from the particular kind of tree bark and not the water in this case. Highly consider that any lakes, ponds, or swim may have these little creatures in the water. They used to use old Christmas trees as a habitat in lakes and streams here. Yeah, Bex, I see people fishing in these waters. I think I've seen canoeing as well, but I haven't seen anyone swimming, which is a good thing. Big Gamey, there are 57 Gucci's. And Chad, if anyone in the community would like to add their very own Gucci sound clip, you can do that. On the Discord, there is a channel called Say Gucci, and you just upload your sound clip and say Gucci. There are people who want to do a couple of more Gucci's on there. Beach Teen! Hi, Beach Teen! Welcome in, Beach Teen.
You Gucci? I'm Gucci.
That was Dabs! The lovely Debs. The lovely Debs. So, Chad, this one's empty. Let's move on. And there's not always...
That's pretty Gucci, eh?
That was Yogurt Garrel. There's not always a correlation, Twitch Addict, with the kind... Like, if you have nematodes, will you not have tardigrades? So they're not necessarily competing for the same stuff. Yeah, this is one. Hi, No Doubt Big Trout. Welcome in. How you doing today? Why are you ducking, Smikes? Oh, that's why. That's why. That's a fair reason to duck, Smikes. Because Gwen hasn't submitted. I know, Smikes. Eventually Gwen will submit one, but we are not ready for it, Smikes. So here we go. There's one here. There's a nematode right there. You're doing good, no doubt. I'm glad you're doing well. We're doing Microscopy Monday, and we're looking at we're gonna be looking at some ancient amber, but we're hard hunting for tardigrades. Instead, though, we were coming across this really cool nematode. And nematodes are cool because you can see the reproductive organs. So that little tube inside the animal that is coming across sometimes visibly when she's not flailing, that is the reproductive tract. And there's both male and female sex organs there. They are hermaphroditic. They will occasionally produce a male if they're undergoing environmental stress and they need essentially a genetic refresher to introduce some variability to their gene pool. Yeah, that would be a good one, I think, to have. Oh, hold on, chat. There we go. Okay. Not the same, but close enough. Yogurt, you have access to that emote. Yogurt Garrel, you have access to an Ellie Elegans emote. Miss Garrel. The lovely Yogurt Garrel. Yogurt Garrel, Chad, She knows these things, but sometimes, chat, sometimes she doesn't like to use them. She doesn't like to use those. You like the wiggle? I do like the wiggle too, yeah. Ray Krieger, she recall the time that I was your mom. Yes, Gwen. That did happen once. That did happen once, Gwen. I'm sorry, Gwen. So there we go. There's one. There's one in here. But not really anything else going on. Again, here's the fun part of live science, folks, is that we may or may not find what we're looking for with respect to the tardigrade. But at least we are still finding other creatures, and we will continue to look across, you know, the coming weeks as well. I'm sure eventually we will find one. Yep, and this one, Twitch Addict, is totally blank. This one just has the debris of little branches here and there. Lure rat with a tuba, yes. Approximately 1% of animals only use. What do you mean, Coffee Rocks? Like tend to have males and females? It's a lower percentage than what you'd predict. I don't know the approximate number, but most animals tend, like, there is a reproductive cost of sexual reproduction, like a negative component of it. See, we've got more swimming on around right now of the nematodes. So there is actually a cost of mating hypothesis, Coffee, that suggests that those animals that have that end up mating have shorter lifespans, and that there is like sexually transmitted diseases, and there is a bunch of negative things that arise as a function of reproduction through sexual means. So having males is not always a beneficial thing. In fact, a lot of times it can be quite the negative thing too. Move on to great. What did you look at there, Gwendolyn P? It's fine, Gwen. It's fine. I love you. I love your face, Gwen. I need to go wool shopping because I have no sense of size or proportion. Would you like to pick a color on Lona's behalf? So, let me grab her blanket, Mom, that she really likes, to show you the color. Is that okay? These ants lions have 10 males only for breeding. Oh, in that case, Coffee, almost animals that have a male, most of them are for the sole purpose of breeding, and they don't actually participate in parental care. There are a couple of bird species. There are, the seahorses, but most males, like, mate and then are gone because their sole purpose is just to produce sperm and try to spread as much genetic information as they can. So yeah, the males tend to usually be poor fathers, Coffee Rocks. All right, I'll be right back. Let me grab little Lona's blanket. This is one of her favorites, Mon. I see her handing the texture of this a lot. I have no idea if she likes the color the most, but she really likes this blanket. So this is one, and then I can show you the other one too. The other one is the same kind of pink, but it has purples inlaid as well. So let me put this back and show the other one too.
Gucci, bless you.
So, this one, Mon. This color here.
Gucci, bless you.
Those are the favorites of her. I don't know if she actually can perceive the color any more than we, like I don't know at what age they start perceiving color enough to be able to like it. But she at least, I can tell, likes the texture. So maybe, possibly, potentially. All right, let us look in the next chamber. Again, a few nematodes.
We'll all be Gucci.
It's all Gucci.
We'll all be Gucci.
That was double Mr. Jimmy B right there. Coffee. The randomizer coffee sometimes doesn't feel as random. But yeah, you can see
Fucking Gucci.
Fucking Gucci. We have right here additional nematodes, but I didn't see any tardigrades, unfortunately, in here, y'all. I am sorry about that, but I do think and wonder we might just have to have had longer in there. Wonder who that was? Mon, I have no idea who that was. I think it may have been Jern. May have been Jern. May have been Jernmon.
Gucci.
But yeah, more nematodes in this one. The other two were blank or just had like one nematode in there. But we will hunt for some more. We will hunt for more later on. What I think I might do, we'll see if we get to it tonight. We might not. But I'll soak some additional pieces of bark while we're looking at pieces of amber. So we might find something later on. This is just like, give us a little bit of something extra to look at later in the evening if we so choose, and we run out of amber to look at, which I can't imagine will be a thing, but we might as well be prepared. All right.
Gucci.
Jern mad swearing spree says. So anything with Jern swearing is always gold, Mon. But we just couldn't do the enough swear. We had to keep it slightly family friendly, Mon. Slightly, possibly. She was horrified. My favorite is the Jason. Mon. The Jason! Which, Mon, when I was at TwitchCon, they were talking about some kind of programming that they were gonna debut at some point in the near future, and that it uses JSON code. Gwen, exclamation point Jason, if you want to trigger it. And Mon,
Jason.
I messaged Jern because I just couldn't stop saying the following phrase that goes afterward. Jason's not a code. That might be why their code didn't work, Andrew. That's maybe why the live debut didn't work. But Mon, I even messaged Jern. I was like, you know who the heck is here? It's Jason. Jason the rat. It's the whole thing, Mon. All right. So I am re-soaking some more pieces of wood for if later in the evening we decide to look for some more samples, we can. But no pressure if we decide not to. Is there a target temperature of the best hands of catching tardigrades? So, Coffee, when we got really lucky, it was early summer that we were catching them. Pix! Thank you for the follow, Pix Stitch. Welcome the heck in, Pix. It was early summer, Coffee, that we did several runs and caught a fair number last summer. Later in the summer, was it like September or October is when we started losing like luck on that front. So I'm assuming it's probably not quite warm enough yet. Nice job, tactics. But soon enough we'll be more likely to catch them. I think it's still fun because we can look at things in the pond water and find some kind of microorganism and micro life as well that we wouldn't have seen otherwise. But hopefully we'll get lucky again. If not now, then soon after. But I'm looking forward to being able to catch some more of these little critters. All right. Let us do the first piece. So we have two pieces of Cretaceous amber. I know Gwen is very, very excited, chat. Gwen was like, I'm sick and tired of us looking at pond water. I want to look at some amber. What's really cool about this first piece, I haven't seen what's inside, but I will tell you right now, is that the clarity is quite high. Usually Cretaceous amber is much dirtier, but this looks really, really nice. Let's measure the size as well of our critter with our lovely ruler, King Charles. I'm sorry, Gwen, you were not tired. You're right. Gwendolyn P., you were. Gwen, that was the far less angry. That was the far less angry one, Gwen. Let's try again. There we go. That emote and command was made because of Gwendolyn P, chat. Because of Gwendolyn P. Gwendolyn P was like, I really want to rage. And I was like, yes, you got it, Gwendolyn P. You got it, Gwendolyn P. For you, Gwendolyn P, anything, Gwendolyn P. There we go. Look at that. All right, so we haven't considered what this is yet, but this little critter is a few millimeters in size. I think if we turned it on inside, it'd be probably 1, 2, 3, 4, 5, 6 millimeters. 6 millimeters in length, maybe, depending on if we stretch out its legs. Maybe it would be 7 or 8 millimeters. So it's a small spider, but it's a pretty nice looking one, I think. So we assume it's a spider. Let's actually also save the image. Alright, 4/3/23. 4/10/23, right? Yeah, 10. Why did I say 3? hold on, y'all. I made a boo-boo on the folder naming. There we go. And saved. Alright, awesome. Let's go back to the video file. Hi, Crux. How you doing, Crux? No sorry, Crux. No sorry. What happened? Nerve. Alright. Try to do the zoom out stage. Nor, how you doing, Nor? No, Nor. There's no obligatory, as Dev says. Bitties here. Nor, how the heck are you doing? Welcome on in, Nor. Thank you for the biddies, Nor. Nor, how are you, my friend? We just looked at some pond water, and we found some nematodes. And now we're doing some ancient amber. This is Cretaceous, so it is... Gwen, the range of this age is... I mean, it's plus or minus 10, so it's anywhere from 100 million to 80 million. Microscopy Mondays. No, thank you, Crux. Crux, you're one of my favorite days as well. Crux, this piece was alive during the time of the dinosaurs, which I think is really cool. Doing well. Just got home after a double. Oof. Nor. Welcome home. Glad to have you here, Nor. Nor, also, thank you for making me laugh. Every time we're over at the Mocos. You always make me chuckle, Nor. There we go. That's a nice little zoom in here. Oh, look at that. This is an amazing piece. Sorry, y'all. I keep adjusting. There we go. That's nice and centered. Yeah, it's really clear, and we can see so much additional detail here. There we go. Alright, let's adjust the lighting a little bit more. There we go, look at that. Boom. So, what's nice on this one, too, is I feel like, because we can see the sensory bristles on it as well. You can see the mouth parts too, which is pretty neat. Hi, Kai. Welcome in, Kai. Kai, how are you doing today, Kai? Kai, we're looking at our ancient amber right now, and this is Cretaceous amber, alive during the time of the dinosaurs. Nor! Thank you. Spider bits, your friendly neighborhood spider bits. Nor can it swim from a web. Yes, it can, because it's a spider. Look out, world, it's a spooter pig. Thank you, Nor, for the spider bits, Nor. Appreciate you, my friendo. See the mouth parts pretty well, too, and the hair is super remarkable, the res that we're able to get here. Look at that. That is remarkable. I don't think we've ever had as good of a spider, chat. That's definitely a winner right there in terms of the image. Let's also color adjust and see if we can check out a little bit more of what its color might look like. Ooh, there we go. It clears it up nicely. So we've told the software of what is white, and so we've change that yellowy hue into the white, and so there's still a little bit of a hue around it. You can see a little bit of the orange color. So that is because there was some compromisation, or the exoskeleton was compromised here. It started to leak out some kind of hemolymph or insect blood. So that's what these little orange, like, almost looks like. It's like almost a watery discoloration that we're seeing. We can try to find the side of like where it was compromised. My guess is probably the abdomen has busted. And sometimes under the pressure, the legs will also bust. But it's a very interesting pose that the spider has. What we've talked about in the past, Chad, and I think it's really cool to be able to see this, is that this animal wasn't already dead when it was preserved in the amber to begin with. So the way you can tell is if the animal had already died and then got caught up in the resin is that it will actually be shriveled up. You can see this spooter is not shriveled up, but fairly active and like almost like in an aggressive posturing. Or it's even tooting, as Nor says. And so that's a really interesting feature that we're being able to see. So this spider presumably was walking around and it got caught in the resin, and that's what ended up killing it, which is, I think, really, really cool. So we get to see much more of the actual, like, the biology of what the spider would look like in the wild. And again, the mandibles here. Tough to make out, but there are the eyes and the mandibles right underneath. Maybe we'll do some of the manual zoom. By the way, look at the legs. Let me change the exposure. Daria Lynn, thank you for the lurk, Daria. Just check out the res that we're able to get and the clarity of those little fur hairs on the leg. Y'all, that is super cool. That is really, really cool. Oh, and I didn't take a zoom out picture. I forgot to take a zoom out. Hold on. All right, let's take an image like this. Where's Cliff? I'm not sure where Cliff is. He's off being amazing, Gwen. You know, he's very musically gifted, talented individual. He's probably saving the world through the power of music. Now let's zoom in on the mandible. Hi Tech 2674, how you doing? You try? No, Cliff, you're breathtaking. Yeah, check out the mandibles, Gwen. Tech, how are you doing today? Gwen, Cliff even made a mask of an ant. It's his profile picture on Discord. It is really cool. It is really, really cool. Gwen, this sample is beautiful. On top of it, Gwen, I want to take a look at the other side and see what the other side looks like. Gucci indeed, Kai. Yep, that profile pic was made by built by the amazing and breathtaking Cliff Alster McLean. I think Cliff, that's how he introduced himself to us. He's like, oh, ants. I've made an ant mask. Like, you have, Cliff? Like, yeah, let me show you. And it was like, ooh, that's really cool. That's really, really cool. Cliff rules, chat. Gwen, let's look over on the other side of it. And see what the other side looks like. I'm going to change back our color. There we go. I know, right? Coffee, I think what gets me, too, in addition to just being really, really beautiful, it's also 90 million years old, so it's also one of the oldest samples that we have. In addition to being really beautiful. So there we go. Oh, let me scoot it over. It's a very, very small piece, so like any little movement is like not at all little movement. There we go. We'll zoom in, of course. I just like to show y'all the... Here we go.
Gucci.
I think it's quite Gucci, Mon. Mon, 3 guesses as to who that was. 3 guesses as to who that was, Mon. Also, I feel like this is the perfect moment to do this redeem.
I've always wanted to say that.
It's just such a clear piece. Definitely been... No! That was Jern! That was Jern, Mon. Mon, you know Jern. You know Jern, Mon. I think Jern has like 6 Guccis. That was Jern. He goes Gucci for one of them. It's a very different flavor of Jern. It's a very different flavor of Jern. All right, let's zoom in. My sisters. Aw, Kai. I hope they enjoy. Dakum is jumping out of my hands. Yes, Tech. I'm so excited that you won it as well. And because Tech, can we say why you were so excited? I don't want to dox you. But chat, Tech won a Kevin pin today over the Moko stream. And he was really excited because he was like, oh, this is a really cool pin. And then Derek and Dev were like, oh, his name is Kevin. And he's like, Tech's like, wait a minute. My name is Kevin. I need this pin. And he summoned the power of Kevin. Oh my goodness. And sure enough, he won. Oh, I'm so sorry, it's just wiggling all over the place. We also, Tech, we have a bunch of Kevin animations here. For example, if you went here, hi, Silly V! Chat, if you're not checking out the amazing Silly V, what are you doing with your life? Amazing artist, extraordinaire, and programmer. We love Silly V here. Chat, go check out the lovely, amazing Silly V! Nothing silly about them. I mean, it's fun. It's like fun, silly, but it's not like silly, like, oh, it's like just pure talent right there. Pure talent. So, Tech, when you won, Kevin was hyped. On top of that, Kevin also wanted to throw sparkles at you. But, Tech, I don't know if you celebrated Easter, but Kevin did. And he's very silly because he throws the eggs at people. But you know what? It ends up being alright because he likes St. Patrick's Day. He also gets yeeted sometimes. He likes St. Patrick's Day. He also celebrates...
Five Kevins.
Halloween. He very much likes Halloween, I think. But on top of that, he also likes Christmas. And he also steals treasure. steals treasure. Kai, your nieces are laughing. Kai, this Kevin, I tell you, and he throws turkeys at people too. And he dances. So Tech, you're a little bit famous here. Or some astronomy show there. So some of those did have the 3D animated Kevins there as well. A little bit, just Grimly, just a little bit Grimly, just a little bit. I think that's all the random Kevin animations that we have. There's gonna be more. I'm planning one for Star Wars month that should be a lot of fun. But I think that's all the Kevins for now. We'll always have more Kevins, but you know we have fun. But welcome the heck in to lovely Kevin as well. You are now
one of us, one of us, one of us, one of us, one of us.
So this is the backside of the spider. You can have Kevin's Tech. Do you know why we named him Kevin? It is the most insane logical dump of things. It was just Lita and I were like, I made him, I drew him out. I think I did the sketch first, and then I did the cartoon, and then the 3D model, I think was the order. And all of a sudden, we look at him, we're like, you know, he looks like a Kevin. I don't even know what that means, but that's why he's named Kevin, because Lita and I were like, yeah, he's like a Kevin. No idea what that means. No idea. Absolutely no idea. Carlo, this is an automated message. Subscribe is alive and would like to return to normal human mode following a home invasion, unexpected family. Is everything okay, Carlo? Is everything okay, Carlo? Hi, Sweet in the Bell. Welcome in, Sweet in the Bell. So this is the backside of our spooter. We'll go ahead and do some color changing. You're dying to art? He is. It says clear. It says clear, chat. He's dying to art. Okay, hold on. Let me bring him back. Let me bring back Carlo. There he is! There's Carlo right there. Welcome back, Stream Scribe. Chat, remember the person above my head doing live letter art is the legendary, the breathtaking, the amazing Stream Scribe, a.k.a. Carlo. Please go drop him that follow. Remember, he is live Monday, Tuesday, Wednesday, Thursday, Friday. Meme Monday, Let's Talk Tuesday, Word Art Wednesday, New Things Thursday, Free Day Friday. Today, or this Friday, is going to be a massive giveaway day. He's giving away even a whole PC. I don't mean public communication. I don't mean personal communication. I don't mean protective custody. No, I mean one of those computer things right there. I don't actually know what PC stands for in terms of the computer world, but it's neither here nor there. It's neither here nor there. That's what he's doing. He's giving away a whole streaming computer on Friday. This Friday, folks. Make sure to follow him and be there. Or be square. You don't want to be square, folks. Personal computer? Smikes, I was thinking for a moment portable. And I was like, no, it's not really portable. Would you like to do an impromptu It's up to you, sir. Up to you. What would you like to do? What would you like to give away, Carlo? Is there any rules for the giveaway? Hi, Michael R. Hassler.
Say what?
Hand lettered. I would love to. I would love to. mailed to one winner. Chat, would y'all like to do a raffle for a piece of hand-lettered art that we mail to you? I think that would be fun. Chat, or who's up for that? Who's up for that? Are we up for it? Spectre's up for it. Hi, Spectre Flight. Okay. Let me get the keyword going, Carlo. We use Nightbot here to get that going. We use Nightbot. Hold on now, chat. Let me go ahead and get the keyword set up. Alrighty, exclamation point raffle! Exclamation point raffle to get up on in there, folks. Exclamation point raffle to get on in there. Carlo will make you a piece of hand letter art that he will mail out to you. So it is going to be done on a piece of paper, like the calligraphy behind it. And he will mail it out to you. Carlo, are we doing international shipping on this? Is there international shipping? So folks like Debs, if Debs wanted to enter Carlo, could Debs enter? Yes, okay. International is also Gucci, folks. Anywhere in the world, for a piece of hand-lettered art by the lovely stream scribe, a.k.a. Carlo. That'll be an absolute hoot, Carlo. So exclamation point raffle to get up on in there, folks. We'll keep the raffle open for a few minutes before we'll close it, and we'll roll it for the party people. So make sure to go ahead and type in exclamation point raffle. Exclamation point raffle, folks. And we'll roll it as we always do. Remember, those who, let's see, one moment, I will send a new link so we can, we'll see it, we'll do it live on the cam, chat. We'll do it live on the, live above my head, chat. We'll do it live. We'll do it live with the lovely Carlo. So make sure to get up on in there, type exclamation point raffle to get on in there. Reason for offering is the dying iPad that needs... Totally fair, Carlo. I understand. I'm so sorry about the iPad, sir. I'm so sorry about the iPad. You know, they get loosey goosey sometimes, Carlo. The iPad. But what are you gonna do? What are you gonna do, sir? We need to get you like a long charging cable that's just like always around and near it. RN So's, how you doing, RN So's? Welcome on in, RN. How you doing tonight? Guys, go check out the legendary amazing RN So's, also part of the Moco Loco stream team. How you doing, RN So's? Welcome on in. We're checking out this piece of Cretaceous amber, 90 million years old, arrived during the time of the dinosaurs. RN, we also have the lovely stream scribe, aka Carlo. He's gonna do for us a piece of hand-drawn letter art of whoever wins. And if you win, he will mail it to you. He will mail you the piece of letter art. And it is anywhere in the world is available to do. It's just exclamation point raffle to get on in there. Reminds you of the honeypot ant. Yeah. So especially from this angle, astronomy show, it does, like, if you're like, you know, it looks a smidge like it's 3 legs and 3 legs, but there's that you know, the hidden one right here. And then the upper abdomen here too. Of course, RN So's. Always RN So's. Always RN So's. So that's one of the features that we're working on today, is looking at one of these pieces. So remember, y'all, if you want exclamation point raffle to get in on the raffle for a piece of hand-done letter art by the lovely Carlo that he will mail out to you, chat. Exclamation point raffle to get up on in there. If you've already typed it, you're already in. But we'll keep it open for just another minute or two until Carlo is ready. Until Carlo is ready to do the do the poll, as it were. Getting close, chat. It's that time. When he says he's ready with the link, we'll do the raffle for it. Let's go ahead and color correct the image. Oh no, he didn't have to send a new link at all, chat. Look at how fancy Carlo is. The sir is the most fancy. Remember, if you go over to his stream, refer to him as sir or doctor. Oh, he's also an Aveeno hand model, folks. He uses that Aveeno hand moisturizer. I use the same one because it's not oily. It's not oil-based, so you can still use nice things. Gold Bomb, same thing too. The Gold Bomb. Look at that. Look at how handsome his hand is. Like, it's that Gold Bomb moist hand model, in addition to being the most handsome man in New York. Carlo, give me a thumbs up if you're ready. If you're ready for the raffle pull. Thumbs up! All right, chat. Are you ready, folks? Are you ready for the raffle pull? Whose name are we going to do in a good old-fashioned 3, 2, and 1? Lara Gale! Lara Gale, you are the winner, not of a sticker, but of a beautiful piece of hand letter art. Carlo is doing beauty right in front of your eyes. Oh my god, I can't even focus now. This stream is sponsored by Bond, Gold Bond. Shaken but never stirred. That's true, BitGamey. You don't shake it. You don't shake it at all. If I have an honorary PhD, am I allowed to be called doctor? No, Spectreflight. Absolutely not. Absolutely not, Spectreflight. Absolutely not. I mean, you can always ask, but I would say no on that front. Look at that. Look at how wizarding fast this man is, chat. Look at his hands, too. Beautiful hand model right there. Carlo, the stream scribe, live letter art above my head. I'm just mesmerized, chat. I can't even do the microscope. You are an ordained. Yeah, I know you are an ordained minister, Spectre. Who are you marrying? Isn't there someone that you're marrying soon? Some fancy pants person that you're marrying sometime soon? I felt like Spectre like Zedman. Yes, that's what I thought. That's the assumption as well. If I have read all the Dr. Seuss books, can I be called doctor? No, BitGamey. So reading them doesn't make you doctor. That just, you read a book by a doctor. You ever get around to, oh, he hasn't agreed, Spectre? I thought he'd agreed. I'm so sorry. Carlo's art is mind-blowing. It's so amazing. Dak, I'm right. Remember, Carlo was in the wedding business for a long time, and can you imagine having your wedding invitations and your seat signs and all your signs for the wedding done by Carlo? It'd be the most beautiful wedding ever. Like, everyone would want to take home their little name cards and everything. He made them all, right? Grimly, it would be the best wedding. What if I watched all the Dr. Phil? Oh, then you get all your degrees retracted. BitGaming. You lose all your degrees. All your existing degrees, whatever you've had, whether it be GED, MD, PhD, Masters, Bachelors, I don't care what it is, you get all of them retracted, BitGaming. Yeah. It's a big game. I don't make the rules. I don't make the rules, BitGaming. I just enforce them. Chat, before y'all came in, and y'all triggered a lovely hype train, we were looking at some ancient amber, 90 million years old. And we have a spider inside of this. We're looking at the front side of the spider as well. Huge amount of detail from anywhere from the fur, the mandibles, and so much more. Oscar, thank you so much, Oscar. It is a spooter, Music Girl. Music Girl, how is Music Girl Monday, by the way? Chat, if you don't know, Music Girl sometimes streams on our friend, it's Jimmy B93 stream. On Mondays. Make sure you're checking out the lovely, amazing Jimmy B93. Very handsome, very kind. IRL friend of ours. Stressful? I'm sorry, Music Girl. Music Girl, you have to tell Jimmy to get it under control. He can't be adding stress to your life, Music Girl. I don't like that, Music Girl. Overcooked is too stressful. See, that's why I can't be playing stressful games anymore. I'm too old and have too little limited time with video games to play stressful stuff. That's why. New glasses, hard to get used to them. Are they working any better? Athena Cat? Welcome back, Observer Mama Bird. Welcome back. Hi, Jimmy! Oh, you didn't realize it was... I mean, isn't the description of it stressful? You're supposed to cook as fast as you can. BitGamey, you want a steaker, by the way, BitGamey. BitGamey, you want a steaker. BitGamey, you want a steaker. Hi, Tommy Diplodocus. Thank you, Tommy. Tommy, we had a hype train. If we complete a level 5 hype train, I always put on one of the ferret onesie. Yeah, Big Gamey, we assumed you got so scared that you dropped everything and that you couldn't tell us what happened, Big Gamey, or which one you would like. I'm sorry, Big Game. Big Game, which one would you like? You have the Twitch words variants. You can spin the cookie wheel for the golden. You can do the magnet. Well, actually, I think you've already gotten the magnet. The band members. All the things. We can't do that, but there's other stuff like wind and... Here's the back end of our spider. Again, we're leaning towards this being possibly a wolf spider, given how hairy it is as well. Yep, that is Carlo Ugamugga. If you type !scribe, I can do it for you in the chat. That gives you Carlo, all of Carlo's information. His Twitch page as well as his Twitch schedule is there as well. And remember, he is on his partner Poosh, so go check him out because we want Carlo to be Twitch partner. But he does a collab stream with us on Mondays during Microscopy Monday. He always does live letter art above my head. And he does people's names in chat. He's also been doing fancy SciAnts things as well. I don't know what we did to deserve the fancy SciAnts things, but they've been quite lovely. So here is the back end of the spider. And actually, chat, remember how we saw that red haze in the amber? If you look all through here, you see how this is the abdomen of the spider. That empty region right there, and here that we can see through in terms of the lighting, that is more evidence. That is more evidence that there was a pop here. So presumably this animal's abdomen did rupture and there was some of the insect hemolymph that leaked out. Very visible on this image here. You see there is some of that like orange haze around the animal. That, again, that transparent region is probably where you would have gotten that pop. It shouldn't be that color. We'll take the image here. It shouldn't be that color, like transparent, if it weren't for that event happening. You guys making Athena, my glasses made me dizzy for a long time as well. I had to reset for a while. I couldn't look above and below them because they would make me so dizzy. It was terrible. It was terrible, Athena. But eventually you grow used to them. I also like now taking them off Athena Cat and just resting my eyes, which I remember. I remember the olden days when I didn't need to rest my eyes, Athena Cat, where I just had a nice thing to be able to do it. Sit back and relax and listen. Love you, Athena Cat. Thank you for hitting that command, by the way. So, chat, this is our wolf spider, preserved in Cretaceous amber. Aga, your piece is done. Aga, if you want to take a screenshot to save a beautiful copy of the piece, feel free to do so. Yeah, beautiful piece, right, Bit Gaming? All right, I'm gonna make a little sticky note and write down what this sample is. So this is Cretaceous, a Cretaceous spooter. And a pretty good spider as well. So Tommy Diplodocus, this is from the Myanmar region of China. So that's where these 90 million year old samples come from. We've gotten lucky and been able to acquire a few of these. So we actually looked at 2 90 million year old samples last stream, and we've got a second one to look at today as well. Screenshot shell version so far. I love it. Oh, good. Thank you so much for loving it, and thank you for the kind words. I love it. Catalog, aka the stream scribe. All right, let us look at the next one, folks. The next one it's another Cretaceous sample. Who's next? Who would like your name done in live letter art? Who would like your name done in live letter art? Chat, say me in chat. What wasn't Debs? Debs, were you not on the menu? Docket? That's a better word. Better word, Debs was on the docket a couple of weeks ago. Debs wanted like 3 weeks ago done. Claire's asking for one now. So, Carla, I'll leave you to choose, Carla. I'll leave you to choose, because Debs has been waiting for a few weeks. We did do some pond water, Athena Cat. Yeah, we found some nematodes. But, Athena, don't you worry, Athena Cat. Athena, hi, first-time chatter! Hey, you, get off my cloud. Welcome on in. Welcome on in. Welcome the heck in. Hey, you, get off my cloud. If you missed Athena Cat, all of our stuff is being archived now on YouTube. Look at its little feetsies! Look at its little feetsies! By the lovely and the amazing Debs. They will be on YouTube. Debs cuts out the intro and the outro and the raids, and so that way it's just pure SciAnts that you can look at. And, you know, you'll be able to see we found a bunch of nematodes today. We were searching for tardigrades today, Athena, and so we were soaking pieces of tree bark. I'm going to do it again in a little bit. We're going to look again in a little bit. Look at its little feetsies! Look at its little feetsies! Because I didn't have any to look at earlier on. So we soaked some of these tree bark samples, Athena, and
Wonder Woman, Wonder Woman.
we didn't find any, Athena, tardigrades. Instead, we found a bunch of nematodes. Now, what I did was, when we switched over, Athena, we switched over and decided to do the amber, and then I'm soaking more tree bark now in the water, and we'll see if we find any more samples of, well, we'll probably find more nematodes, but I'm curious as to if we'll get any more any tardigrades at all. It might be too early in the season, Athena. And then, Athena, over the weekend we had a freeze, and that freeze led to what I assumed, like, that's why I didn't bother getting a lot of pond water, because it was just, the freeze was intense enough where I'm like, we need to be careful. We need to be, we need to be careful on that front, right? So, no, so tree bark, Athena, that's where, tardigrades are. So that's where, with the lichens and the mosses on the trees, if you crack those off and you soak them in water, you can usually find a tardigrade if it's in the right time of the season. We didn't get any today, so I'm soaking some more, and so after we're done with this piece of amber, we will look at those pieces under the microscope and see if we've been able to identify any more tardigrades. At least that's the plan. If we don't get to it, Athena, it's also okay. Tonight we're going over to the lovely Nerduino at 11 p.m. Eastern. He's going to be chatting with me on his stream. We're going to go over a little bit earlier so I can grab some food before it's time for Nerduino. But that's the plan anyway. Ooh. Chat. It's another piece of Cretaceous amber. This critter was alive. 90 million years old. Common housefly. How big is it? How big is it is the good question, Cliff Astor McLean. Cliff. Alright, Cliff, we'll take a picture. So, Cliff, it is 1 millimeter, 2 millimeter, 3 millimeter. Hold on. It looks like the width of it, if with its wings out, it's about 6 millimeters. And then here, the body of it is maybe 2.5 millimeters, if we're looking at that. There are no houses. What is a house, Gwen? What is a house if not a container for something? Got it. Did Balint drop the amber? Yes, Balint. A cave. Let's see, cave fly. Reverse Uno to you, Catallo. Whatever, if you dared say any niceties, right back at you, sir. New show called The Ark uses the tardigrade science to keep alive and continue across space travel. Yeah, they had that one, and Athena, there was in one of the new Star Treks as well, they had as well down that front. Is there a way to keep moths? So Big Gamey, get a yellow light. Big Gamey, at Home Depot, there are bulbs that are yellow. If you have that yellow light, it doesn't attract moths, and it's visible to you. Like, you can still see just fine in there. That's what it's not as appealing, Bit Gamey, because it is a yellow light, but like, you won't get any moths. Hi, Econ Greg. Chat. Another amazing science streamer doing usually astrophotography is the amazing Econ Greg. Please go drop that follow. Econ Greg, how you doing today? Welcome the heck in. Chat, go check out the lovely, amazing Econ Greg. Econ Greg, we love your content, my friend. How are you doing tonight? So yeah, Quoth. The wings look really beautiful. And Cliff, there are... It looks like to me, Cliff, that there are 4 wings. So this one right here looks like a doublet. Right? The way that line is coming down, that doesn't look like an error. That looks like a wing right there. And then here, Cliff, is where I'm having trouble identifying the second wing on this side. Taking a break, good to see you rocking it. Glad to be here, Econ Greg. Glad to be doing the science content. We did some pond water lichen microscopy earlier, and now we're doing the ancient amber. This is 90 million years old. Absolute treat to be able to see something like this underneath the microscope. Let's actually zoom in. I will take an image like this. And remember, folks, all the images are uploaded on the Discord. So if there's something cool that you like seeing, just join the Discord, and it's an entire channel is dedicated to seeing like the microscopy images. Okay. Dacum, early bedtime tonight. One side client was good luck, Dacum. Dacum, sleep well. Have a great evening, and thank you for hanging with us. We love you, Dacum. So as Cliff points out, because there's 4 wings, it is not a fly. There's a lot of fuzzy texture to it, so we're looking at one thing that has a lot of sensory bristles. There we go. Flea Flow, Fla, NMD. Take a couple of pictures. So the flea NMD, I think the wings are far too big. And yes, fleas tend not to have wings. Oh, it could be a flu. Oh, I get you, NMD. Hi, Portalgeist. Welcome in, Portalgeist. The wing veins are beautiful on this creature, though. Look and see it. Even the wing veins have fur on them. Alright. And actually, chat, really quickly, I'm going to go ahead and take off the ferret onesie because we've hit our 30-minute marks on it. So I will be right back. I'm going to take off the onesie. I'm going to use the restroom real quick. While I'm doing that, I'm going to leave you all in the capable hands of the lovely moderators, and gotta love the stream scribe. I'll be right back, folks. Don't go anywhere. say? Andrew knew what he was gonna say. He said, Andrew knew what he was gonna say. AMD 96 AMD is how you say hero.
Don't you like me a little bit?
Ew, I don't even like you a little bit.
Huh, I heard you like me a little bit.
Well, maybe just a little bit.
Gucci. Bless you.
Rush art.
The spell is broken, and we live again. I've always wanted to say that. It's science.
I'm disappointed that no one used the new command today. In the chaos, the ensuing chaos while I was gone, no one used the new one.
I've always wanted to say that.
Quath the Raven, thank you for using the 3D. Thank you for using 3D.
Education and some inspiration spitting from the heart. I know that I got the chemistry spit, the science, it's my art.
Thank you, Big Gaming. So it's Athena Cat. It's exclamation point 3D does this. Antonio 3D printed a Kevin. 3D printed a Kevin. How dare? How dare? And hi, Digital Deck. Welcome in, Digital Deck. How you doing, Digital Deck? Good to see you, Digital Deck. So, chat. Let's look at a couple of other features of this animal. So we're going to flip it over. Actually, let me see if I can change the color a little bit. There's some... Here we go. What you can see is actually there's some color that has leached out here, and it probably has a popped abdomen. So there's some hemolymph that's come out. And so... No, it's still really beautiful to see. Here. There we go. I'm just really digging the detail in the wings, and actually that's what's giving me a clue as to what this group of insect this might belong to. Athena, we are... I was telling the party people about this the other day. So starting in May, on our Science Art Sundays, we're going to have a theme. And it's going to be a month-long theme. May is going to be Star Wars. So there's going to be all Star Wars science art with our characters doing Star Wars-y stuff. June is going to be Marvel. July is going to be DC. So we'll have a Wonder Woman, a Superman, a Batman, all those fun ones. Haven't fully decided on what August is going to be, but other ones that we're planning on is we're doing Disney, Harry Potter. Big Gaming, thank you, thank you, thank you, Big Gaming. And there are a couple other. Oh, we'll have classic cartoons, video games. So a bunch of things coming. They'll be themed by month. Wednesdays on our art days, we'll just do random stuff that I'm wanting to do.
We Gucci.
So those will be some extra bits there. So that I think will be a fun one to do. That tactics, I hope that'll be Gucci. Question: so you don't have an official BRB? So we do have an official BRB coffee. It's this one here. And that's actually playing one of the Antonia video game sounds or songs. You never gave it to me, Carlo. Sir, I never got the updated one, my friend. But we are also going to have one. No, I never got it. Never got it, Carlo. Never got it. Otherwise, I would have popped it right out. We're also going to do one Coffee Rocks that's going to be the stage where the theme song plays, but instead of having the characters in the theme song playing, it's going to be a sanitation cast of the leafcutter ant, and she's going to be cleaning the stage. And we're going to have the outtakes reel playing. So you'll get to see some of the silly mistakes or goofy things that we've done on the stream. Including but not limited to feetsies and me making random noises that were put together by the lovely NMD96. It's pure gold. It's pure gold! Coffee Rocks. But yes, the theme song, as Coffee points out, that we have is done by the legendary Thug Shells.
Look at its little feetsies! Look at its little feetsies!
Go check out the lovely and amazing Thuggy folks.
NMD 96! NMD is how you say hero.
And NMD96, obviously, because she is on her partner push. Okay, let's image from the other side. I have a suspicion as to what family this bug belongs to because of the wings. There's a whole group of ones. Trying to get more theme songs without paying for it? Can't. Coffee. Absolutely not. You can absolutely. I would never ever, Coffee Rocks, have any blame. Any ever blame on your Coffee Rocks. I don't want the theme song to get too old, which is why we leave a little bit of a. That's why there's that channel point reversion on there as well. But I'll give you all a hint. So I think based on the structure of the wing, I think it falls into the lace wing family. And again, it's a big part of it is because of its wings. So that's what I'm leaning towards, that it's a kind of lace wing. And lace wings do have 4 wings. But let's take a look at the other side of this critter. Ooh, actually, this is quite a cool shot. Hold on. Let's switch back to the regular colors. Okay, so there it is. Let's adjust the lighting. There is a bubble. But y'all check that out. Check that out. Look at how clear the eyes are. And then we have the mouth too, and then there is the antennal structure right there. Now, if we have it all the way zoomed out, you can see there's multiple focal planes that things need to be in. So we'll need to be image a couple of times. Was that secretion a secretion form bubble. So optic, my guess is no. So see, optic, the bubble is quite large. Like, it extends here from like the thoracic region all the way down to the abdomen. Usually, optic, when you put these critters into any kind of resin, like modern day resin, independent of it being tree sap or not, it just, the wings are very hydrophobic, as are a fair number of the other body parts, because that's what the sensory bristles help do. And so you almost inherently will be getting bubbles. So my bet is that it's not from an excretion, but just from ending up in the resin. And given that this, again, doesn't have the, like, the shrunken down body, like the pulled in body, it was probably alive at the time of being caught in the resin, which is unfortunate for this little critter, but good for us because it looks cleaner. Let's go ahead and take an image of the face, which I will need to hold on. We'll do a zoom out first. Of course, this will have to do some image stacking on this eventually, because you can see there's a bunch of them. I'll be here listening. Love you, Athena. Athena, good luck with the glasses, ma'am. Bowel movements, they could optic, but usually when it's captured, there's just no bowel movements. It's not like with humans where after you die you poo. Instead, it's just, more than likely what would happen, optic, if there's any kind of bowel movement, it's from a rupture, so that you would rupture part of the abdomen, and that would probably lead to discoloration. Now, will we be able to see that on this? I think the bubble might be masking our visibility on it. Okay, there we go. A nice little zoom in here. Okay, that's max zoom for the face. Cliff said it does look a little lacewing-like for the face there. Golganak, how is there no worm time? Or tapir time? Golganak, I tell you, that pup time is all kinds of loosey-goosey, Golganak, with what it ends up actually deciding to do. Tragic end, but not forgotten 90 million years later, almost immortality. I mean, in a way, right? That it does make you all the more powerful, right? There's a hands time? Oh, no! I don't like that. They're all sad. Why is hands time sad? Shame. Truly a shame. Truly a shame. But there are the eyes. There is the mouth part. There we can see the antennae quite well, and see like even little fuzzies on there. We're seeing a fair bit of definition on this piece. Let me adjust the color and see if we can see a clearer face. It sometimes helps. It's a little bit better, I think, for what we're trying to do here. Yeah, I think this coloration is better on it. No, whatever, what y'all think. What I'm surprised by. So, do y'all see the color of the eye? You see the color of the eye right here, and how it's not, and how it's white. So that white color, that's usually indicative of a mutation. There it is. That's the new bee right back. God, that's beautiful, sir. And it's a real bee on there. Godalo, the stream scribe. So the eye here. Hi, Arjun Live. Welcome in, Arjun. How you doing, Arjun? I see you too, Quoth the Raven. What I'm surprised by, it's a very light-colored white eye. And that light color white eye is usually not found in the wild. In the wild, a white eye means that the animal is near blind. So, and in the lab we've shown that. So you can mutate eye color in a fruit fly. It's called the white gene. If you mutate that, it's backwards. So the gene name is named after
96 Adam Day is how you say hero.
Is named after
You're in the game for smart.
what happens if you mutate
I just
it.
don't. People. I don't people.
So if you mutate the gene, it ends up becoming white-eyed. That white gene. So normally, when you have a normal copy of the white gene, you have red eyes. When you mutate the gene, you have a white eye. And given, I don't think this is a function of us color adjusting, even here with the amber color, you see how there's not really any red here whatsoever, any kind of color in the eye. I do wonder if this, it's not that they were domesticated, Quoth, because it's, a 90 million year old sample. My suspicion is that the red that we're seeing here, I think it was leached out. The eye color leached out, which does in fact happen, Quoth. Despite my state, I'm still just a bug in scope. Pattern Juggler, how you doing, Pattern Juggler? So yeah, this right here, given the sample, and Carlo, did you hear my Monday story, sir? Because Carlo has the Monday meme right there. Jimmy, I love you, Jimmy B. I woke up this morning, Carlo, because Lita's like, she's waking me. It's like 6:30. It's my turn to take care of the baby. And I woke up in a shock, and I was like, is it Monday? Like, I did not think it was Monday. I thought it was some other, I thought it was a weekend day. And she's like, yeah, it's Monday. And I was like, oh God, okay. And I jumped up and I was ready to go. Monday's a construct, but it means I have meetings and whatnot. And I really thought, I was so tired. I felt like I got hit by a train. I had a headache, and then I was like, okay, it's clearly Sunday. It's fine. And no, it was not. It was Monday. It was Monday, Carlo! So this I agree with your meme right there. So, Quoth, it's not that it stared into the sun for too long. You can actually do this with samples in regular, everyday labs, Quoth. What you do is you take, a red-eyed animal like a fruit fly, and you put it into a vial of methanol or ethanol. And that's what you preserve the animal in if you're about to do dissections later on. It's just, oh my goodness, Carlo, hold on. Let me fix the, we changed our width. There we go. Sorry, Carlo. The sizing changed. There we go. Okay, now you're Gucci, Carlo. Yeah, so it's already dead. But Quoth, what ends up happening is if you leave it in the methanol, so the methanol is a preservative. And if you, no, don't, you I have to delete Carlo's message because he's trying to apologize and stuff. This is from Myanmar, Cliff. This is from China. So what ended up happening, Quoth, is if you put the animal into a tube. Debs! Sleep well, Debs!
Not tethered by earthly limitations.
What does that mean?
Come.
Debs, sleep well out of your face, Debs. It's your fault. You nerfed me. Yes, that's totally fine. You can say it's my fault, Carlo. That's 100% true. Most things are my fault. I will 100% accept this. I'm a monster. I try hard, but I'm still a monster. Sleep well, Debs. Thank you for being amazing. We love you, Debs. We love you, Debs. I'll talk to you tomorrow, Debs. Yes, Carlo, you're getting nerfed, Carlo. proving that I am, because I made those sounds with Baby Lona sitting next to me, and I'm just going, "Wah!" It's a whole thing. It's a whole thing. You kind of just made up of plastic stuff that they make football helmets out of. You know, Carlo, you're not too far off the point, because there has been evidence to demonstrate that in our water now there are microplastics, and these microplastics can actually get caught in your bloodstream and even lead to like blood clots and stroke. So there is a higher number of these particulates in your bloodstream than had been even 20 years ago, just because of how much plastic we're putting in there, which is very, very unfortunate. So, Quaff, so the reason you put it in methanol is to preserve the animal. If you want to do an immunohistochemistry, or if you want to do a stain, like to image where is the DNA located in the animal, or what is the outline of the cells, or what does the brain look like all these other extra things, Quaff, to do that you have to do immunohistochemistry. That first step is you have to do a preservative. You have to put the sample into a preservative, like methanol, ethanol, or formaldehyde. If you leave it in there for too long, meaning, so when we did the all protocols that I've done is like overnight at 4 degrees Celsius. Why 4 degrees? It slows down the chemical reaction. You can do room temperature at for 2 hours, or overnight at 4 degrees. So it's just usually if you're doing work at the end of the day, you pop it into some methanol, and then you throw it in the 4 degree. Yep, so this is a natural preservative. Exactly, Quaff. What ends up happening, Quaff, is if you leave it in for too long, which is on the order of like 2 days, some of the colors from the eye will start leaching out and in. So if you dissect a fruit fly head after a few days in the formaldehyde, the brain becomes bright red, because all the pigment has leached into the brain. And it's unusable. You cannot image anything on that brain. Same thing with the rest of the body as well. So it's called over fixation, because Quaff, when you fix something, it is cross-linking stuff together. So let's say there's a protein floating here in 3D space, and near it is the gut, and near it is other cells and whatnot. With the formaldehyde, it freezes everything in place, and there's, between cells there's liquid, but there's also this thing called the ECM, or the extracellular matrix, and that's what's holding together everything inside the body. The formaldehyde, the methanol, the ethanol cross-links, which means it essentially connects everything together and freezes everything in space and time. Overfixation is when all those cross-links become too much and stuff starts to crack. So your DNA inside the nucleus starts to break. So you can't do any kind of really imaging or any kind of processing of those samples after it's been overfixed. Same thing with like the color might leach as well. So I can't prove this to you, Quaff, but my suspicion is, given that these are white eyes are not found in the wild. That is the lacking of the pigment gene, which means the animal is blind. If it were in nature, it's not going to be blind. It would not have made it to adulthood because it needs to see to survive. My suspicion right here is that some of that pigment leached out. And that's what we're seeing surrounding here as well. That doesn't mean that the abdomen couldn't have ruptured, but that's just one thing I'm thinking as a side product here. Methanol, ethanol, isopropanol. Isopropanol is another fixative to NMD. High concentration of isopropanol. They do rhyme quite nicely. So would that happen if you get distracted? Quaff. The reason I know this, Quaff, is so the person who trained me when I was an undergrad, his name is Nathan T. Mortimer. He is an amazing friend of mine. He was the best man at my wedding. Okay, that's how much of a best friend he is. he taught me all my, like, how to think science. And he taught me how to do brain dissections and immunohistochemistry on fruit fly brains. NMD, I called him a captain. Because I feel like it's a pirate name. NMD. So he taught me how to do these dissections, and we were trying to do it on a species called Drosophila seychellia. So, Quaff, that is a close relative of Drosophila melanogaster, which is most commonly in the lab. What did we end up doing? So, we did the melanogaster, and then we wanted to do another one, the seychellia, but it was a Friday. And Quaff, we really wanted to not come in over the weekend. So, Quaff, we put it in formaldehyde at the end of Friday, and we're like, we'll just do it on Monday. It should be fine. Because why should it not be? Quaff, we checked on Monday, and all the red eye pigment from Drosophila seychellia had leached into the brains, and Nate told me they're called angry heads. I don't know if he made it up or not, but I just believe it at this point. And so they are they're just all like the whole head was red. It had leached into the brain. If you dissected it out, it was red. We tried to do the experiment anyway, and it failed totally. We tried because Nate was like, he told me it would fail, but he was like, let's learn about why it would fail anyway, because it was good practice to get into dissections and for me to do the whole protocol. And then we did it again, and it failed again, but it failed because the antibody didn't work, not because we did the protocol wrong. Would it make a body after embalming nearly impossible to be exhumed and retested? So, the DNA can survive coffee, but if you're looking for anything beyond sequencing, you're not going to be able to do it. So if, like, after you mummified someone and embalmed them with formaldehyde, and, like, you know, you take out the canopic jars for the organs and everything, we couldn't do anything like high-C, which is an analysis that measures the 3D structure of chromatin, because the formaldehyde would have totally ruined that. Same thing with our own, like, modern-day preservation techniques as well. Does that make sense, Coffee Rocks? Let's go over and get an image of the lower body here as well. Pure science, I messed up. How? What do we learn? So, Quaff, I wish I had more mentors like that. In graduate school, Quaff, I wanted to learn some new techniques. And I was told, if you want to learn new techniques, watch a YouTube video. Quaff, I don't know about you, but I don't know how to do a Western blot by watching a YouTube video. Because a Western blot is a way to measure protein levels in a sample, and there's a lot of different things that you have to do to get right, and like there's a lot of places you can mess up that unless someone is telling you who has messed up on it, a YouTube video is not going to tell you, right? Because YouTube's like, do this and this and this and this. And so I never learned how to do it in that lab, so when I went to start my postdoc, then we started learning, like, again, it was the same idea. It was the exact same, it was the same idea. I was like, okay, and I learn now. And then all of a sudden, instead of being told that I can learn again, someone, like, the person who was supposed to be teaching me got even angrier because they were not about learning.
96 ain't them they. It's how you say. Here.
And so it was a whole thing, chat. It was a whole thing. It was a whole front on that, and we weren't allowed to learn at all, and it's very frustrating. So, Quaff, my much more is, like you say, let's play, let's learn about how we do this, versus just having there to be something extra happening, right? Like, of how to do the next step. Does that make sense? That's how I would learn. And whenever I had students that I had to teach, that's what I ended up doing. Where all the students, I taught them how to do different techniques. I made sure that they learned why things didn't work. I didn't set them up for failure. We just went through all the possibilities that could go wrong. And sometimes something new went wrong that I hadn't encountered. And, you know, my student would be like, oh, shoot, like, his thing went wrong, I'm disappointed. I'd be like, you know, don't be disappointed. We've learned another way that it could go wrong. Which is fine, because now we're not going to make that mistake again. And if we do, then we won't make it a third time. Right? Like, it's fine. Like, you learn by making mistakes, not by having everything perfect from the beginning. Hates Bio right now because their labs have to do a paper in person. And to be in person, at the same time, he has learned better. So Quoth, in my intro bio in college, we had to run a polymerase chain reaction in a gel. And my gel was blank. Not just mine, my partners and our 2 other partners across the table from us. Our PCR was blank. And we couldn't redo it because there wasn't time, and the woman who was teaching, she was like, oh, I can't have you come back because I'm busy. And so, but we had to write a whole paper on it. And even more breathtaking is that because we had to write a paper on it, she's like, just write why it failed. And we're like, we don't know why it failed because there's nothing no one told us why. And she was like, it's fine. Chat, I think Cliff figured out exactly what this is. First, folks, let me show you a modern day lacewing. Wait, okay, let's take a look right here. This is the abdomen. I just wanted to show you all that real quickly. It does look like there is some emptiness in the abdomen. My goodness. There looks like there's an emptiness here, so it does look like the abdomen has been drained at least somewhat. I'll take a picture. Probably from some kind of rupturing. But it looks quite nice. Let's take a look at what Cliff found. Real quickly. So this is the lacewing that I identified that based on the structure of the wings, this is a modern day lacewing. And you can see it has the right number of wings. So it has the 4 wings. It has fuzzy wings, and it has a lot of these crisscross wing vein patterns that I think look very similar to what we would have had in modern day. Now, here's what Cliff found.
In dreams,
Music
one is
girl!
not tethered by earthly limitations.
What does that mean?
Come.
Sleep well, Music Girl. Sleep well, you breathtaking soul. I love you, Music Girl. Music Girl, please give Jimmy a big pinch on the booty from us. Please give Jimmy a big pinch on the booty from us. This is what Cliff found, and my goodness, Cliff, I think you've 100% nailed it. So this is a long proboscis neuroptera from Upper Cretaceous amber of northern Myanmar. Tell me that doesn't look very similar to our sample. Look, the eye, so I know it's an artistic representation. They got it right. It's not a wide-eyed. It does look like there was a proboscis there, but look at the wing structures as well. Hi, Jern! Same fuzzies and the same wing veins as well. Look at that. Cliff, I think you totally nailed it. Hold on, there's other images here too. I mean, I'll be a monkey's uncle, chat, which I know is a silly phrase to say, but I wanted to say it. I'm sorry if it came across as awkward. It's fine. Everything's fine. It looks very similar to our sample. The eye, obviously, it looks like the eye is a different color, so preserved a little bit better. We did not get a good enough image of the face to get this. I think, if anything, the proboscis is underneath here. Awesome. Thank you, Spectre Flight. Thank you, Spectre. Spectre, you want to do one more time? Exclamation point, Spectre. Spectre, do you want to do one more time for old time's sake? Or I can trigger it for you too, Spectre. Thank you, Spectre Flight, for your honesty, my friend. Appreciate the heck out of you. Where did I put this? Aha. Thank you, Spectre Flight. No worries at all, Spectre. No worries at all. Thank you for the heads up and letting us know. So let's go back to the image. Again, we're not getting the full information on the proboscis, but I'm not convinced that it's not underneath the bubble. See, this right here is what we're missing. This structure right here. But otherwise, I mean, if you look at the furriness of the wing, all of them looks very much on point. Cliff, I think you hit the nail on the head, Cliff Alistair McLean. Very nicely done, Cliff. Cliff, it also makes me chuckle that all the resources were in Korean. So, Chad, we found a very beautiful Cretaceous sample today. Cliff, this is awesome. Cichridae, known as sponge flies, are a family of winged insects in the order of Neurotrema. Approximately 60 living species and several extinct species from their fossil record. Thank you, Cliff. Hi, Mom Boss Lady. You're just in time, Mom Boss Lady. You're just in time. We're going to be wrapping up in a couple of minutes. We're going to be going over to Nerduino tonight, who is then going to be doing a interview chat with me about SciAnts and SciAnts. Both SciAnts and SciAnts we're going to be doing. So, we're going to be rating over in just a few minutes. We're going to review what we've seen. We're going to review what we've looked at today. Yeah. Did I eat today? Would you count a diet bar NMD96? Would you got a diet bar NMD96? Oh my God, no! It's very well done, NMD. Okay, lace, wing, and Cichridae, awesome. Mama Don't Play. It reminds Carlo. You've heard that song. Mama Says Knock You Out. How do you spell Nerduino? Portal guys, I'll give you. Well, actually, one of the mods. Can y'all shout out Nerduino? Give a Fancy Pants Pinky Out shout out, please and thank you. Mama Said Knock You Out is actually about NMD96. Let's see. Yes, Portal guys, we will in just one moment. Let me record the sample, or if one of the mods can do the Fancy Pants. There we go. There's Nerduino right there. That's where we'll be going to in a little bit. Okay, so this is Long Pro Boscus Neuro Terra. Awesome. Sweet. Thank you, Cliff. So, we will put this sample away. Chad, this was a very good haul tonight. We did 2 really beautiful Cretaceous pieces. Gwen, unfortunately, for the time being, that's it for our Cretaceous samples. Not to say that we can't acquire more, but there are gonna be we don't have any right now. We don't have any right now available, Gwendolyn P. So we did go through our Cretaceous samples this week and last week. Doesn't mean there won't be more, but we still have so many of the other amber, of the the Dominican amber. There's tons of Dominican amber that we still have to go through. So I think we're gonna be okay. But Gwen, I'll keep my eye out for more Cretaceous ones. Usually the Cretaceous samples are more expensive, so I try to limit in what we are able to get because it's much pricier. But you know, we figure it out. We figure it out, Gwendolyn P. All right, I'm gonna put Baby Alona's pillow back on this chair. Because, chat, when she joins me tomorrow, she needs to have her pillow ready. She has her little back brace blanket here, so she can sit here, her little booty here, and her feet, and then she looks at me, and we just hang out and play and work and whatnot. Because you know, we get a little... We're very silly, Willy, Baby Alona and I. It brings me the most amount of joy to make my daughter smile, chat. It's mind-boggling. I thought what I was seeing related to Mayfly because it looks... No, there's no quitting, Quoth. Appearance and if they look similar is a very good first step into looking and seeing what they're related to. It can be quite difficult to delineate further down simply because we don't have any genetic information on them. Chat, let's quickly look and see a quick review of today. Hi, Risto! A quick review of today's pieces. We did identify some nematodes today in the pond water. We're not going to check the additional samples that I've been soaking because we gotta round out the evening, as it were, so we can go over to Nerduino so that I can grab a little bit of dinner, and then if we do that, then you know, I'll have some time for some food before we go over on Nerduino stream, which would be a nice thing to do.
The spell is broken.
And Quoth, if you want to trigger the other tardigrade, Quoth, you have access to the other tardigrade. If you type !hero, you can trigger the other tardigrade. Go. So I know, NMD. We found some nematodes today, and they're quite active. Actually, again, it was really difficult to get any good imaging because of how active they were, but we're getting close. We're getting close on getting a good picture of them, chat. I think maybe the zoom outs were probably the best ones, but I'll post them on the Discord either way. Then we had, as Coffee Rock says, 2 excellent, mind-numbingly cool pieces of 90 million-year-old amber today. We had a spooter, the spider, and see all the different features of the spider that we looked at. Mandibles, the back end as well we looked at. Really, really cool. And then we had this lovely lacewing. Again, I'm telling y'all, the wing veins are so beautiful on these. I love the wing veins and what they look like. Absolute stunning piece. And the face, ooh, and Cliff Alistair McLean coming up clutch and identifying it. Very beautiful work. Everybody, chat, I think we had a great stream today. How about y'all? You think we had a great stream? I think we did. I think we did, chat. Hi, Lilith. Chat. Big thank you to Carlo the Stream Scribe, who finished just on time. NMD, get a screenshot. NMD96. Guys, make sure to check out the lovely and amazing, breathtaking Stream Scribe, a.k.a. Carlo. Chat, please go follow Carlo on his partner Poosh. On his partner Poosh, folks. Go check out Carlo the Stream Scribe. He is live tomorrow. Yes. Oh, yeah, we did talk about the leaf cutter and Quoth. We need to watch that documentary sometime soon and dissect the science behind it. If anyone missed anything today, have no fear, because the YouTube page is there. The lovely Debs goes through, and Debs removes the intros, outros, removes the raids, and gets you distilled down the SciAnts content. So make sure to subscribe to the YouTube page, chat.